السلام عليكم جميعا. Welcome back everybody. السلام عليكم everybody. Welcome back to our activity for today. أرحب بالجميع وأتمنى لكم التوفيق جميعا، وأتمنى تكونوا استفدتوا يوم أمس، وبمشيئة الله احنا حنبدأ المحاضرة وتقريبا على الساعة 3:15 بناخذ بريك، وبعدها إن شاء الله بريك للصلاة، صلاة العصر، وبعدين الساعة 4 نكمل مرة ثانية زي أمس. يعني تقريبا احنا جدولنا مشابه اليوم أمس إن شاء الله. رابط التحضير بعد المحاضرة هذه إن شاء الله، فنتوكل على الله. طبعا زي أمس يا إخواني، إذا في أي سؤال عن طريق الكويشن اند انسر بوكس أو عن طريق الشات بوكس، وفي أقرب فرصة نجاوب. كانوا الزملاء أمس بيعلقوا أنه أحيانا المحاضر بيوقف شوية وبيكمل، لأنه هو المحاضر يا إخوان بنفس الوقت يقرأ الشات بوكس ويقرأ التعليقات، وبيحاول يرتب الأمور مع بعض. الاتندنس شيت موجودة فوق يا إخوان. الشيء المهم أنت ما تطلع من الزوم، والمحاضرة مسجلة. التحضير مرة واحدة في اليوم، اللي يحضر مرة واحدة خلاص كافي. فإذا طلعت مثلا كافي بريك أو شيء أو لأي سبب كان، بتخلي الزوم شغال ما بتفصل. مهم هذا. طيب نتوكل على الله يا إخوان علشان نلحق نكمل نلحق على وقت الصلاة. Our first topic is about E. coli diarrhea. During the 1940s and 1950s, series of outbreaks of diarrhea in hospital newborn nurseries were reported, in which etiologist agents appear to be E. coli identified by serotype. These strains became known as E. coli epic, enteropathic, enteropathogenic E. coli. Since the second international symposium of EPIC in 1995, strains have been renamed typical EPIC T to distinguish them from another not well categorized E. coli strains known as atypical EPIC. Since 1970s, enterotoxigenic E. coli strains have been shown to be important cause of diarrhea. Etic strains are being, which is the enteroinvasive E. coli, being introduced in the developing regions such as the United States, strains which has been identified in patients with dysenteric illness, clinically indistinguishable from that produced by Shigella strains. التحضير يا اخوان موجود فوق، ما يحتاج تكتب اسمك يا اخوان، سجل في التحضير الرابط اللي فوق يا اخوان. Enterohemorrhagic E. coli, EHEC, or Shiga toxo-producing E. coli, STEC, strains were shown to cause colitis with dysentery and to produce hemolytic uremic syndrome primarily in children. While the most common serotype, STEC, is 157 H7, many serotypes of Shiga toxigenic E. coli can cause illness. Our food supply is currently an important source of STEC infection E. coli strains, showing entero-aggravative attachment to the epithelial cells and referred to entero-aggravative E. coli, were identified as cause of persistent diarrhea in infants living in developing countries. AIDS-associated diarrhea and traveler's diarrhea. These so-called diffusely adherent E. coli, DACC, are currently being evaluated in the case of diarrhea. The pathogenicity of these strains is currently uncertain. DAC will not be included in this review, and in this will review information available about EPIC, ETEC, EIEC, and STEC, and EAEC. Little is known about actual incidence and mortality of illness seen in the diarrhegenic E. coli infection. It is necessary to rely on outbreak data for information obtained from prevalence survey. An increased mortality reaching 16% has been seen in newborn, particularly if illness is complicated by prematurity. EPEC strains appear to differ in virulence characteristics of E. coli 0111, which was shown to be associated with striking clinical illness resulting in common occurrence of fatalities. ETEC strains produce self-limiting disease in healthy persons from the industrialized countries, traveling to most regions of the developing world, but severe cholera-like illness can occur. Mortality is unusual with ETEC. There are no accurate data defining expected AIC mortality rate, but reported studies suggested the death would be unusual. Uncomplicated STEC infection, including dysentery disease, show low mortality rate in comprehensive community outbreak. When complicated by hemolytic uremic syndrome, STEC diarrhea is associated with mortality rate of 10%. Enteroaggressive E. coli is an unusual cause of death, with fatalities occurring primarily in patients with advanced AIDS.
Since E. coli diarrhea, other than that caused by STEC, is not reportable disease, data and morbidity must come from survey information. Sporadic cases and community and hospital nursery outbreak of epic diarrhea have been documented in nearly every country where appropriate studies have been carried out. 3 types of studies have been done to determine importance of epic illness, survey of relative frequency of recovering epic strain in population with diarrhea. A study of newborn nursery outbreak of epic disease and surveillance of epic strains during short periods in communities under study. Survey of ETAC, EIEC, STEC, and EAEC diarrhea have been restricted to short-term studies of diarrheal cases and outbreaks in several regions of the world. I think we will have a break يا اخوان for عصر break. نطلع البريك يا اخوان الصلاة العصر ونرجع كذا على الساعة 4 مشيئة الله نكمل المحاضرة. طيب يا اخوان المعذرة يعني we have to accommodate معلش يمكن بعضكم ما يجي مناسب لوقت الصلاة بس we have to do it this way للأسف ولا نقدر نطول أكثر من كذا. طيب نراكم على خير يا اخوان. Welcome back, everybody. We'll continue our lecture for today. ما يحتاج يا اخوان، حضروا عن طريق الرابط يا اخوان، عن طريق الرابط. التحضير عن طريق الرابط يا اخوان في الشات بوكس. Isolation and identification of organisms. Methodology of E. coli isolation is readily available in microbiology text. And any diagnostic laboratory problem and diagnostic laboratory that perform bacteriological isolation can isolate E. coli with a proficiency. Biochemically, the aerogenic E. coli are not distinct from non-pathogenic E. coli present normally in intestine. E. coli strains that cause illness can only be differentiated from normal flora strains by demonstration of virulence property. This has also presented a problem for the routine clinical bacteriology laboratory because demonstration of E. coli virulence properties or E. coli pathotype require methodology generally available only in research and reference laboratories. This diagnostic problem has limited what we know about epidemiology of E. coli diarrhea, although recent advancements in molecular techniques have greatly improved our diagnostic capabilities. Complete serotype E. coli strains require determination of both O and H antigen types. There are approximately 164 O groups and 57 H groups. Generally, 5 E. coli colonies per patient sample need to be tested for both toxins. Immunoassays have been developed for detection of OST, including radioimmunoassay and enzyme-linked immunoabsorbent assay, ELISA. These tests correlate well with results of sucking mouse assay and require less expertise, as early in 1982 DNA probes were found to be useful in the detection of LT and ST encoding genes in the stool and environmental samples. More recent advances in ATEC detection have been made, including PCR assay, which are sensitive and specific when used directly on clinical samples or on isolated bacterial colonies. A useful adaptation of PCR is a multiplex PCR assay in which several PCR primers are combined with the aim of detection of several different diarrhogenic E. coli pathotype in a single assay. Following a study with the multiplex PCR, various reaction products can be differentiated by a single agent assay. Presence of virulent Shigella or EIC strains traditionally has been verified by testing the Guinea-pig models. In the assay, heavy suspension of bacteria was dropped into the conjunctival sac or Guinea-pig 7 days later, observed for presence of virulent keratoconjunctivitis, confirming the strain capacity to invade epithelial cells.
DNA probes and PCR methods are now available to test directly the presence of genes encoding for invasiveness. Volunteer experiments have been conducted to establish the virulence of epic strains. Epic strains have been defined a specific serogroup of E. coli that being epidemiologically incriminated as a cause of diarrhea, but do not produce conventional enterotoxin, are not invasive. The focal attachment of strains to small intestinal epithelial cells has been shown to be associated with pathognomonic lesion at the site of attachment, described as attaching and effacing histopathology with microvillus destruction, intimate adherence of bacteria to intestinal epithelium. Pedestal information aggregation of pluralized action at the site of attachment. لا تحضير امس ما اقدر والله افيدك يا اخي، تسال الادنى. في الشات بوكس ما اقدر والله اجاوبك يا اخي، ما اعرف صراحه. اخوان بالنسبه لي يسال يقول لك انا احضر بحساب مختلف بالزوم مختلف عن رابط التحضير، اضيف معلوماتي الصحيحه ينفع؟ الافضل يكون رابط الزوم، حسابك في الزوم نفسه اللي في رابط التحضير عشان ما يصير فيها دوبليكيشن. The chromosomal factors are carried in the locus of enterocyte effacement and include type 3 secretory systems, E. coli secretory proteins, translocated intimin receptor that is inserted into the intestinal membrane as a novel organism receptor, and AA gene encoding with intimin. LE also carry tyrosine kinase, which together with double calcium lead to formation of actin-rich pedestal of aDNA, and ETAC strains produce one or both of the toxin enterotoxin. The frequency of epic in stool of pediatric diarrhea is higher when hospital cases are compared to outpatients. In other reports with highest frequency of epic disease, three geographically distinct areas were involved in one outbreak, and the three epidemic consensus tracts attack rates were 73, 118, and 55 per 1,000 among non-white infants less than 1 year of age. Intestinal carriage of the three different household groups was 18.3 and 0% respectively. Kissinger et al. Carried out a prospective examination of EPIC and defined geographic area in Illinois and Indiana. The population morbidity rates was 0.116%, with age-specific attack rate of 0 up to 2 years old group was 0.98%, and for 6 up to 11 months old was 2.4%. Epic strains have been shown to be responsible for both epidemic and sporadic diarrhea, whilst ETEC has been reported to cause low frequency of diarrhea in the United States and Canada. Recently, outbreaks of illnesses have been identified. ETEC strains may produce epidemic of diarrhea, such epidemics that have been associated with the consumption of food or water contaminated with ETEC. Although outbreaks have been documented in hospital nurses. These organisms were the most frequent isolated pathogen among Mexican children with diarrhea. Latin American poses high serum antibody titers to LT, while North Americans pose relatively low antibody titers. In a study carried out in rural Egypt, the incidence of ETEC infection was 1.5 episodes per child per year, accounting 66% of first episode of diarrhea after birth. EIEC has been also shown to be a cause of endemic diarrhea in Brazil among travelers to developing nations. Epic strains have predilection for spread within hospitals, particularly in newborn nursery population. Although organisms spread within community, and the hospital probably plays a role in transmission of community members. ETEC infection in the area of endemic infection tend to cluster in warm, wet months when multiplication of ETEC in food is most efficient. Intrafamilial secondary transmission of O157 H7 have been documented to be 10% during community outbreaks. Furthermore, TAPEC continues to be an important agent responsible for infantile diarrhea in developing countries. Endemic ETEC diarrhea has been shown to be common only in the developing world. ETEC diarrhea has been reported to occur with a high frequency in Latin America. ETEC diarrhea have been reported to occur in high frequency in Latin America and South Asia, and EAEC strains occur worldwide as the many other bacterial enteropathogen. EAEC infection rates may be most common in developing countries where enteric disease is hyper-endemic. EPAC diarrhea occurs year-round with summer time peak incidence, the time of year probably does not greatly influence disease rates. In two semi-tropical countries, Mexico and Morocco, ETEC infection appear to be most common in rainy summer months. Since E. coli diarrhea is not a reportable condition, we do not have adequate information about epidemiology and optimal control and intervention procedure. No specific information of the control of EIC is available. STEC infection is best prevented by avoidance of undercooked beef, especially hamburger. There are no principle for prevention of EIC diarrhea other than avoiding consumption of contaminated food. The most important factor in treatment of diarrheal disease is the maintenance of fluid and the electrolyte balance by administering oral dehydration fluid. The antimicrobial drugs of value index of rifampicin, chloramphenicol, and azathioprine. In studies ETEC and EADC diarrhea, a variety of antimicrobial agents have been used with success in preventing illness among travelers from low risk to high risk areas. The most active drugs are fluoroquinolone and absorb rifampicin. Of these, only rifampicin can be recommended for general use as chemoprophylactic agent based on efficacy, safety, and minimal concern about development of resistance of non-absorbed drugs. Further, the group in which you consider rifampicin prophylaxis is the group of immunosuppression or where underlying illnesses could predispose to complication of diarrhea develop. No immunizing agents are available for APEC, AIC, or HAC infection. Efforts are currently underway to develop vaccine against ETEC. The impetus of this twofold: first, ETEC strains are major and predictable causes of diarrhea in international travelers. Military and state departments employ traveling to developing country regions among children living. Second, ETEC immunity occurs naturally. In ETEC endemic areas, the longitudinal study of protection of natural ETEC infection children revealed ETEC infection produced almost 47% protection against new infection. An orally administered cholera vaccine made killed ETEC whole cells, and binding subunits of cholera toxin is currently available in some areas of the world for prevention of ETEC diarrhea. LT patch vaccine effectively prevented moderate to severe travelers' diarrhea in phase 2 clinical trial. The ATAPEC disease, the most important tissue to resolve this current importance of this group of organisms, which are rarely studied today. In ATEC diarrhea, the most pressing problem centered around the development of assay efficiency identify STLT, CFA in the routine diagnostic laboratories. I think that was the last slide. Any question يا اخوان؟ اي سؤال؟ طيب thank you very much all of you. هو معلش يا اخوان الصوت يختفي شويه لانه اقرا الشات بوكس واقرا الكويستشن انسر، فاعطوني يا اخوان بس يعني شويه يعني فرصه. Which candidate, Dr. Hayam is asking, which candidate patients can use prophylaxis or travelers diarrhea? First of all, is it available on the market or not? It's the issue availability. Not sure that it's available everywhere. The second question, if you are going to an endemic area, you can utilize it. The second issue is the area that you are traveling to. Is it labeled to have effective immunization schedule that you are going to take or not? And last but not least, the vaccine, as we described in the presentation, it's not 100% complete. So you have to put this in consideration, of course. I hope that answered your question, Dr. Hayam. طيب we'll go to second presentation يا اخوان. Our topic is about Haemophilus influenzae, and it's a common commensal of the pharynx of human is considered part of the normal pharyngeal flora, in addition to an important cause of human infection. Strains that causes one of 6 polysaccharide capsule types, although if cause invasive infections such as bacteremia, meningitis, septic arthritis, epiglottitis, and facial cellulitis, with type B infections being significantly more common than type A, C, and F. Use of highly successful type B vaccine composed of the type B polysaccharides conjugated to one of the several protein carriers that resulted in dramatic reduction rates of invasive infection in the United States and in many other hosted countries. Strains that possess one of the 6 polysaccharides capsule type A through F, and use of highly successful type B vaccine composed of the type B polysaccharide conjugated to one of the several protein carriers had resulted in dramatic reduction in the rates and strains with no capsule, the so-called non-typable strains, are frequent causes of localized infections such as otitis media, sinusitis in children and adults, as well as bronchitis, pneumonia in adults. While the organism lives asymptomatically in the pharynx, it doesn't cause localized pharyngitis. No vaccine is yet available to protect against infection with non-typable strain. Epidemiological studies of H. influenzae infection requires a system of clustering the causative organism in a meaningful way. A number of typing systems have been used to group strains as alike or different based on phenotypic characteristics such as capsular type, biotype, outer membrane, protein type, or multi-locus enzyme electrophoresis MLEE type. More recently, a number of molecular typing systems have been used to cluster H. influenzae strain based on restriction fragment length, polymorphism, polymerase chain reaction, PCR, fragment size, and DNA sequence of various regions of its influenzae genome. Phenotype-based typing methods, capsule and capsular typing. Classical serotyping requires specific reagents and skilled, experienced laboratory technologists and inter-laboratory variability, and typing results in accessibility high using this technique. Thus, newer molecular amplification methods have been developed that utilize PCR amplification of the capsular gene. A second series of primers such as amplify the type-specific domain, allelic variants of pigs which encode phosphoglucose isomerase cluster with a specific H. influenzae serotype, suggesting genotypic may serve as a marker for serotype. Influenza influenzae have been classified by biotype in the base of 3 biochemical activities. 8 biotypes have been identified. Most clinical isolates are biotypes 1, 2, 3. And while specific biotypes have been associated with virulent strains, this technique is not discriminatory enough to be useful in clustering H. influenzae for meaningful epidemiological investigation.
Early attempts at subtyping Haemophilus influenzae used outer membrane proteins profiles generated by digesting the bacterial outer membrane with detergents or by sonication and separating the outer membrane fragments by gel electrophoresis. This technique was used to track transmission of H. influenzae strains and to compare commensal strains within otitis media strains from the same subject. Outer membrane typing has been replaced by newer molecular-based methods for identifying related Haemophilus influenzae. MLE, double E differentiating Haemophilus influenzae strains, multi-locus enzyme electrophoresis. The largest study of Haemophilus influenzae electrophoretic type amongst encapsulated strains, 280 types were identified. Type B strains were found to fall into two major phylogenetic groups, division 1. Further, MLEE analysis showed that non-typal strains are phylogenetically distinct from type B strains and more heterogeneous. MLEE costly and time intense to perform and has largely been abandoned. The classical typing system to characterize a group of Haemophilus influenzae, capsular typing, and biotyping are limited by their ability to identify only 7 serotype groups. Recently, molecular typing systems have been developed to expand the ability to group H. influenzae. Recently, molecular typing systems have been developed to expand the ability to group H. influenzae. For restriction fragments linked polymorphism-based methods such as pulsed field gel electrophoresis. For polymerase chain reaction, PCR-based typing methods like RABT and ERIC, as well as repetitive elements of PCR techniques specifically designed primers hybridized to complementary genomic DNA. The ideal typing system is capable of distinguishing apparently unrelated strains from each other, but it's not discretely that isolate distinguished unique types. The results of typing by molecular-based methods may differ dramatically in the number of unique patterns observed among various Haemophilus influenzae strains types. Yes, GIS, for each day we have a different attendance link. So each day has its own attendance link. You have to register for each day once. There is no rule of screening test to facilitate early detection of Haemophilus influenzae infection. Positive throat or nasal cultures of Haemophilus influenzae do not predict an association with a specific clinical infection, as many individuals are colonized with Haemophilus influenzae and only a few of those colonized experience disease. Positive throat or nasal culture of Haemophilus influenzae do not predict an association with specific clinical infection, as many individuals are colonized with Haemophilus influenzae and only few those colonized experience disease. Haemophilus gene reside in gamma bacteria and is a number of family Pasteurella, which is also include a genera Acetobacillus and Pasteurella. You're welcome, Gia. Thank you. فاطمة رابط التحضير في الشات بوكس فاطمة. The incidence falls during early childhood, and low incidence in children older than age 24 months. Before the use of the Hib vaccine, approximately 1 child in 200 developed Hib disease by age of 5 years. With the introduction of conjugated Hib vaccine in 1987, the epidemiology of Haemophilus influenzae type B infection changed dramatically. Recent data confirmed that few Haemophilus influenzae virus infection occur in children in the United States, as documented by ABC, Active Bacterial Core Surveillance Program. Because of the variable capabilities of the accurate of serotype analysis in different laboratories, some of the strains of known type may actually represent type B strains. Similar to the data collected prior to introduction of the vaccine, the highest rates of Hep B disease in US children, followed by African American and Caucasian children. The racial ethnic discrepancy in these rates may be related to differences in vaccine coverage rates. The low level of Hep disease seen among children The low level of Hep disease seen among children after introduction of Hep vaccine depend on maintenance of high post-immunization antibody titers among the age group at greatest risk of invasive infection. رابط التحضير في الشات بوكس يا فاطمة. Among patients more than 6 months old of age with Hep vaccine identified in ABC surveillance program, 17% had unknown vaccination status and 28% were unvaccinated. 20% were under-vaccinated, had completed the primary series. The mortality rate among Hep cases reported at 2000 through the ABC surveillance program was 8%. And of these, 80% patients 6 months of age were unvaccinated. Because Haemophilus influenzae live exclusively in human, person-to-person transmission is vital to survival of these bacteria. The organisms are spread via infected respiratory droplets and establish colonization in the upper respiratory tract, nasal and posterior oropharynx. Murphy et al. Demonstrated that Hep and nasal secretions were able to survive up to 18 hours in tissue paper, water, wax paper, and dry gauze. To reduce the potential transmission of Haemophilus influenzae B among hospitalized patients, respiratory isolation precautions are recommended for patients with Haemophilus influenzae type B infection for 24 hours after initiating appropriate antibiotic therapy. Molecular epidemiological tools allow tracking of transmission of Haemophilus influenzae strains isolated from family members, show high genetic similarity suggesting intrafamiliar spread. Molecular epidemiological tools allow tracking of transmission of Haemophilus influenzae strains isolated from family members to show high genetic similarity, suggesting intrafamilial spread. A single genotype of non-typable Haemophilus influenzae may be found in multiple children attending a daycare center, demonstrating transmission within daycare. Environmental factors directly related to transmission of Haemophilus influenzae in human are difficult to identify, but factors associated with sharing of genotype identical NTHI in daycare centers may serve as a marker of transmission. Bacteria factors may also important to person-to-person spread of H. influenzae, but have not been clearly identified. Transmission factors candidates includes absence of adhesions or adhesion variants that lose the ability to adhere to certain epithelial cells. Transmission factor candidates include absence of adhesion or adhesion variants that lose the ability to adhere to certain epithelial cells. With rare exceptions, Haemophilus influenzae exists exclusively in the upper respiratory tracts of humans, where they live as asymptomatic commensal. As such, colonization of the host pharynx is the first step in the pathogenesis of any Haemophilus influenzae infection. Pharyngeal colonization with type B organism and non-immune individuals is relatively rare, with point prevalence rates range from 0.7 up to 5.3%. Cumulative prevalence rates of hip carriage among children in daycare, however, were between 84 and 71% in toddler and preschool age children, with lower rates among infants and adults and school age children. Introduction of conjugate hip vaccine has further reduced carriage of hip in young children, and the appearance of herd effect among vaccinated population suggests that reduced colonization rates extend beyond those children who are immunized. Asymptomatic colonization with non-typable Haemophilus influenzae healthy children is extremely common, with point prevalence rates of approximately 25 to 77%. The wide variability may be related to difference in population studies and the cultural technique. Colonization with non-typable H. pylori begins early in life, increase in frequency during childhood, and then decrease during adolescence and adulthood. Multiple Haemophilus influenzae genotypes are frequently present in the upper respiratory tract,
of adults with cystic fibrosis. Studies from healthy children attending daycare demonstrated from 37 to 43% of throat cultures. A study of chronic carriage in 4 healthy adults over 7 months showed carriage with multiple genotype over time. Risk factors associated with asymptomatic anti-HI colonization include exposure to tobacco, smoke, and attending daycare. Growing evidence confirmed that Haemophilus influenzae inhibit mucosal surface as a biofilm, and several Haemophilus influenzae bacterial factors have been shown to be important in biofilm formation, including presence of hemagglutinin villi and type 4 of pilla and acylation of lipooligosaccharides. Bacteria growing in a biofilm are in different metabolic state than those growing under glycogenic condition. The observation that biofilm growth increases the phosphochloride content and decrease the potency of non-type of Haemophilus influenzae. Prior to introduction of the type B vaccine, type B H influenzae was the most common cause of bacterial meningitis in children in the United States. In addition, type B strains commonly cause epiglottitis, bacteremia, cystic arthritis, periorbital and facial cellulitis, and pneumonia. Haemophilus influenza expressing the other capsular types A, C, D, E, and F also cause occasional invasive infections such as bacteremia and meningitis. Bacteremia and encapsulated Haemophilus influenzae almost always precede Haemophilus influenza infection at distal site. In non-immune children, Hep bacteremia without focus is a relatively common illness. While Haemophilus influenzae bacteremia without focus may spontaneously resolve with no residual clinical sequelae. Early symptoms include fever, lethargy, and irritability, and later symptoms include vomiting, stiff necks, and seizure. Children with symptoms suggestive of bacterial meningitis require immediate medical care to diagnose and appropriately treat this infection. Mortality rates approximately 5 up to 10%, and morbidity includes mental retardation, blindness, and hearing loss, and persistent seizure. In non-immune individuals, Haemophilus influenzae type B is the predominant cause of epiglottitis. Hep is also a significant cause of septic arthritis, facial cellulitis, and infection of infants that are nearly always caused by Haemophilus influenzae type B. Non-immune children under age 4 who are in close contact with individuals with Hep infection are increased risk of invasive Hep disease. The prevention strategy using antibiotic prophylaxis is based on decreasing nasopharyngeal colonization of individuals because secondary cases usually occur within a week of a presentation of the index case. Because anti-HI lack capsular polysaccharides on their surface, the Hep vaccine do not protect against infection caused by anti-HI. A major barrier to use many anti-HI outer membrane components as vaccine agents in their high level of strain-to-strain diversity. Because anti-HI primarily cause infections of the respiratory tract, novel approach to vaccine development, such as a direct mucosal application or modulation of a specific immune response, may be required for a successful HTI vaccine. Polysaccharide of the type B Haemophilus influenzae and PRP licensed in 1985. Based on knowledge that polysaccharide conjugated to proteins stimulate enhanced immune response, the Hep conjugate vaccine differs in their immunogenicity, probably related to the nature of the carrier protein that act as a hapten, and the method of coupling, either covalent linkage or a chemical conjugation to protein to the polysaccharides. The PRBD is no longer used in young infants because of its poor immunogenicity. PRTT and PRPOMP and HPOC have all been shown to generate high levels of anti-PRP antibodies in infants and to be highly protective against Hep invasive infection. Repeated administration of conjugate vaccine elicits booster response and maximizing the level of immune response achieved. The large clinical trials designed to evaluate the efficacy of conjugate Hep vaccine and use different strategies. Classical double-blinded placebo-controlled studies. In contrast, PRPD and HPOC were compared in trials in Finland. A case control study of Hep OC was conducted in the United States of Los Angeles. Because the various studies designed comparing officials and vaccine difficult, the only head-to-head comparison PRPD and HPOC conducted in Finland relatively low infection rates. The poor performance of PRPD among Alaskan natives is likely to relate to its poor immunogenicity. I think that was the last slide. Any question, إخوان? أي سؤال؟ شكرا جودة. أي سؤال يا إخوان؟ Any question? هاني, thank you very much. فاطمة, ريان, thank you very much. سماح, أيام, thank you. عبد الرحمن, thank you. نوف, thank you very much. نزار, thank you. نبيل, نورا, thank you very much all of you. سامية, سمير, معالي, فارس, عبد السلام, thank you very much. أنور, مؤيد, آمل, عبد الله, آمنة, thank you very much all of you. واضحة, thank you. سلطان, نوف, نسرين, هديل, عبير, thank you very much. آيات, بشرى, رقية, سارة. كريم, زينب, thank you very much. محمد, thank you. عبير, قاسم, أنفال, sorry, أنفال, thank you. بشاير, هالة, thank you. طيب يا إخوان, we'll move to the next presentation. There are no questions. Our next presentation will be a combined video and a presentation.
Presented by Dr. Zindel Kalish. Philosophy and practices. He has trained over 1,000 practitioners worldwide in the Kalish method, which solves patient challenges through a proven lab-based approach by means of the Kalish Institute's educational programs. We would also like to let you know that any questions not answered during the presentation will be answered via email after the webinar. So whenever you are ready, Dr. Kalish, take it away.
All right, thank you so much for hosting. Excited to be here talking about my favorite infection, H Pylori. So, from diagnosis to therapeutics, I've treated personally 1,000, maybe 2,000 H Pylori cases in the training programs that I run. I don't know, we see them every day, 5 days a week. So I don't know, that adds up probably to tens of thousands of these H Pylori cases. It's a really interesting infection, and it's often missed in conventional medicine, and we can help people a lot by appropriately treating H Pylori. So it's worthy of an hour of your time to think about this. So this is me, Dan Kalish. I have a rather checkered past. I grew up in Berkeley, California, ended up becoming a chiropractor, ended up kind of crawling up the food chain of functional medicine just because I wanted to get out. So for 30 years, because I wanted to hold that up. But I went on the faculty and I have now done research projects with the Mayo Clinic. Our research project with Mayo proved through Mayo Clinic research that out of the 9 H. pylori cases that we had in our research study, the Kalish method solved 8 of them, 8 out of 9. And that earned me a trip to Japan one year because the Japanese are really into H. pylori. They wanted to learn about this secret Kalish method thing that was happening to solve H. pylori problems. Right now, very excited to be working with Dr. Richard Lord, who's one of the industry leading scientists in our field that developed a lot of the testing that we do, organic acids and fatty acids. And he and I are putting together a whole slew of programs coming up this year, which I'm really excited about. We'll be launching on amino acids and B vitamins and all such stuff. So I'm also still in practice, been practicing for a long time, and really I deal with H. pylori, like I said, every day of the week, and it's something that we should all just sort of reflect upon. And today's talk, I wanted to focus on a practical version. So I'm going to talk for a while with some slides. I'm going to show some lab tests so you guys can see how this really works with the GI map testing, and then we'll open it up for questions at the end, okay? And I'm not going to spend too much time on the slides. We'll skip through these kind of quickly so we can get to the good stuff. We'll look at some labs. But the objective is that you can figure out the kind of people that you should be doing this with, right? Who should you treat H. pylori with? And then who should you not, right? And if they're tired, they have mood issues, they have chronic inflammation anywhere in their body, a lot of these H. pylori people have non-GI symptoms. They don't know that it's H. pylori that's causing their inflammatory back pain problem or their skin rashes. They just don't have an awareness that it's coming from their gut. So I want to talk about also the clear-cut GI symptoms that can manifest. Those are the easy ones to figure out, and understand how chronic H. pylori infections can interfere with amino acid absorption. What does that mean? That's like the most devastating effect of H. pylori, in my opinion, right? Is that, okay, maybe you're not going to get stomach cancer, you don't have an ulcer, but what's happening with the amino acid absorption when your stomach's not working? And how does that impact the entire body? We'll talk about that quite a bit. And then we can look at some cases, right, with GI map testing. So Kalish method, we think about this, and I was trained in this by a naturopath many, many decades ago about how to address GI infections in general. And H. pylori is probably the best example of this stepwise approach, meaning that you want to reduce inflammation, get the lifestyle factors figured out. You want to support secretory IgA levels through an adrenal program before you do all the killing. And that a very common reason why people fail with these programs is because they see H. pylori and then they just want to get rid of it, either with antibiotics or herbs. And it's just not really an effective strategy. You need to think through why it's there, why it's flaring up, and how is stress related to H. pylori, and how can you control for or treat the stress response as the initial opening round in the treatment, get the lifestyle figured out, and then clearing the H. pylori out is relatively straightforward. If you do not address the stress component or the HPA axis component or the adrenal component, and you just go on a killing spree right away, that's not as likely to work. So that's one common, sort of misunderstanding. The second one is that H. pylori can manifest in bladder infections, depression, and anxiety. I've had a half a dozen patients whose main complaint was insomnia. They couldn't, get to sleep and stay asleep, which was resolved completely the moment we treated H. pylori. See that over and over again as a pattern. So, and those patients didn't say my stomach hurts. Those patients were like, oh, now that stomach thing is done, Kalish, I don't know what you did, but I'm sleeping through the night. I'm not waking up anymore. I was waking up because my stomach hurt. I didn't realize it. So this is a widespread effects. And when we talk about the amino acids and how it plays into amino acids, I think you'll really understand why that happens. Now, the obvious ones are gas, bloating, acid reflux. Like that's a no-brainer that obviously you should check a person's stomach and fix the infection. But the less obvious ones, anxiety, ADD, HD, problems with depression, weight gain, fatigue. And again, we'll look at that when we talk about amino acids. And so, one of the things that I think is kind of interesting is just about the human psychology of all this is that there's symptom clusters, and the patients relate to their experience, that they're tired all the time, they have heartburn all the time, they're bloating all the time, but they really are pretty removed from what's actually driving the problems. You know, which in this case, we're really focusing on H. pylori. And in fact, symptoms can change over time. H. pylori could cause heartburn, and a few years later, the heartburn might be gone, and now the person is anxious and depressed. And the symptoms can change of H. pylori so significantly that they don't parallel the original problem. And so there's important questions to ask people along these lines, which are, when did this problem first start? And what else was going on in your life around that time? So that you have a broader understanding of how the H. pylori is played into their overall health picture. That's pretty important because most people forget the original onset of problems, original set of symptoms, and are just dealing with what's in front of them right now. You want to take people back so they understand this sort of bigger picture. And in the way that I teach this in all the mentorship courses I do, is we look at this from a systems approach, a body systems approach. And I've just made up these 3 systems to make it easy. This is not systems biology. That's a whole subset of super smart people at like Princeton and Johns Hopkins that are doing stuff. This is like Dan, body systems. There's 3 of them, okay? Neuroendocrine, GI, and detox. And the idea is that when we're stressed, because we're not eating right, we're not exercising right, we're not sleeping well, we're not meditating, when we're stressed, that stress throws off the neuroendocrine and hence the GI system. And no more is that obvious than with H. pylori, right? Where when you're stressed, H. pylori flares and people get symptoms of what we call an ulcer. And that is a super important thing, just to have some kind of a model so you can explain to people why this is happening and why this is flaring up. Now, on the digestive side, and again, I think this is like the low-hanging fruit, it's just kind of obvious, but belching, nausea, constipation, history of ulcers, constant need for digestive enzymes just to digest your food, having to do some wacky diet like food combining or some crazy elimination diet, that's the only way they can feel good, upper abdominal pain, all these kind of obvious symptoms that tie right back to H. pylori are easy to explain to patients. But where it gets a little more complicated is when it's depression or anxiety, and you want to have some kind of framework for explaining to people, I want to test your gut. I want to run this GI map test. But why, Dr. Kez? Because I have depression and anxiety. And you say, well, you know, I believe that the body's connected, you know, and that if you're not digesting and absorbing proteins, which that protein digestion process starts in the stomach, that's not happening, there's an infection in your stomach, you could have depression because of a lack of amino acids or because of chronic inflammation coming from the stomach. Chronic inflammation blocks the conversion of T4 to T3 that can cause thyroid problems. Chronic inflammation is well known to impact the brain in a very negative way. And amino acid deficiencies, right? So you have a good explanation for why you want to run this test on people that don't have digestive symptoms. People that have digestive symptoms, you know, a little more obvious why you might want to do it, okay? But having those explanations down is pretty important. So then what's coming up, just, you know, at Kalish Institute is we have a mentorship program that's starting now, like right now. If you want to sign up, you can use that $1,000 off discount code, VIP Jan 21. And that's the full-on 1-year training program where you're just like inundated with GI map information, parasites, bacteria, yeast. We do tons of information around GI organs. We do other testing too, like organic acids and adrenals. Very comprehensive training program. And then for those of you interested in just boosting up your business and you don't want to deal with the clinical part, we have a business boot camp coming up in a couple weeks as well, where we just take a month or 2 out and focus on business issues. And I think it's the first time we did this. We're giving a discount on the business course if you're interested. DS 2021. Will get you 20% off the business bootcamp. Okay, so those are our 2 programs that we have coming up if you want to learn more about the way that we work with practices. Okay, so back to our regular scheduled programming here. H. pylori, there it is. That's what it looks like. Messes up your stomach lining, right? It burrows in there and causes problems. Damage to the stomach lining, reduction of stomach acid, common in patients with all these different symptoms. You can get it from food, you can get it from another person. But what I really want to dwell on a little bit here is, okay, so it's messing up your stomach. You can't break down amino acids, because that's part of what the stomach is doing. Obviously, they get broken down further in your small intestine, but this is really important to have normal stomach function. So this leads to the inability to produce proteins throughout the body. This leads to the inability to produce proteins throughout the body, which has body-wide ramifications. You're tired because you can't make hemoglobin. You have unstable blood sugar because you can't make insulin. You're tired and even more tired because you can't make enzymes for pyruvate, so you can't make acetyl-CoA, you can't make ATP. Your immune system's not doing that well because you need amino acids to make antibodies. What are the things that we make out of protein? So again, think about it. You got this infection in your stomach. Why does that matter? Because when you can't get amino acids broken down properly from your food because your stomach's messed up, you can't make antibodies, enzymes, thyroid hormones, FSH, insulin, hemoglobin, a bunch of the neurotransmitters, the list goes on and on. So this is why H. pylori infections are so important to find, even if the person has no digestive symptoms. That's the most important point to take home from today. You got to test everybody. Who do you want to test? Every single patient you have to run a GI map on. My teacher, Dr. Bill Timmons, no longer with us. Well, he's in the spirit world, but he's not on the planet anymore. He died a bunch of years ago. But I do feel like he kind of talks to me. Like all morning long, I was teaching the mentorship classes, and I swear that he felt like he was in the room just like laughing. I don't know, it's kind of funny. But anyways, right back to these old school naturopaths, right? You absolutely have to. You couldn't talk to Dr. Timmons unless you'd done a digestive test. He wouldn't even allow you to sign up as a new patient until you completed a GI panel. All right. Now, they didn't have the GI map back in those days, but I'm sure he would have endorsed it because he's very interested in cutting edge technology throughout his whole career. And so again, you can't even sit down and have a rational conversation with a patient, regardless of what their condition is, unless you have a GI test that you're looking at. Okay, it's that important. And now you're thinking, okay, well, why is it that important? Well, let's go look at if you have H. pylori, let alone all these other infections that are looked for, right? If you have H. pylori, you can't absorb amino acids. You can't make what? Storage proteins like hemoglobin and ferritin. Is that important? Kinda, right? For carrying around your oxygen. How are you going to feel if you can't make hemoglobin adequately? Tired. Are you going to have heartburn? No, you're going to be tired. That'd be the symptoms. Hormones can be proteins too. That's what insulin looks like. It's a bunch of amino acids folded up into this really kind of amazing looking shape. You can't make insulin if you don't have amino acids. You can't make pyruvate dehydrogenase. This is the enzyme that allows us to make glucose turn into energy or ATP. It's an amazing and big and complex structure with all these different proteins linked together. They're all made from amino acids. You can't make that protein without amino acids. And if you have H. pylori, you're not going to have enough amino acids. Immunoglobulins, kind of popular right now because we're going to. You cannot make antibodies if you're missing amino acids. Even one single amino acid out of the 20 that are important will block your body's ability to make all these different compounds, immunoglobulins, stuff to make mitochondrial energy work, stuff to make your insulin okay, blood sugar stable, stuff to carry oxygen. Okay, this is why H. pylori is important, because if you have H. pylori, you can't do all those other things. And that's why people could have a hormone imbalance, a fatigue problem, or depression because of H. pylori, because it's the amino acids that they're not absorbing. Okay, that's just basic science, basic biochemistry, physiology. That's not even a naturopathic idea. That's just how your body works. So prevalence, people get it oftentimes from food. Sometimes you get it from kissing someone that has it. I've had 2-3-year-old children who have it from being cuddled by their moms. It's an easy to pass on infection. And again, that's what it looks like. If you have some downtime and you're tired of reading the news, just Google H. pylori Nobel Prize, and you can read. There's a couple of 3-page, maybe 4-page document that's online that talks about the Nobel Prize they gave out to the researchers that found H. pylori causes ulcers. And that's where this diagram is from. It's from the Nobel Committee's information. And it's quite an infection, but the hallmark of it is it causes inflammation. Sometimes it causes ulcers and stomach pain, but all the time, H. pylori causes inflammation and it blocks the body's ability to digest and absorb proteins. Why does that matter? Now you don't have amino acids. Why does that matter? Insulin, hemoglobin, neurotransmitters, antibodies. You're missing one amino acid. You can't string together those polypeptides. You're missing one amino acid. You can't make those compounds. And that's why people with H. pylori have global problems like depression and joint pain and all these other things because of the malabsorption part of it, right? Not the actual infection itself, it's the results of the infection and how devastating that can be on the body. So guidelines for H. pylori, test for H. pylori, the PCR test that is offered by GI map is excellent, and I use that in my practice on a regular basis. Then once you find it, you want to eliminate it. Then you want to make the microbiome a better place, a happier neighborhood to live, right? And then you want to make sure that you retest to make sure it's gone. Now, testing is important and finding the infection is important, but it's very easy to get lulled into thinking that you got rid of the H. pylori because a person says they feel better, but you didn't. So you want to double check with a retest as often as you can. Patients are not always going to go for that. It's a little expensive. Maybe they don't want to do the retest. Maybe they feel so much better. They don't want to do another test. But I really try to convince people to make sure that this thing is gone. And then there's all kinds of different lab testing available. Endoscopic exams fail to catch it a lot of the time. The breath test is designed for acute infections. We're looking for chronic, so that usually misses it. There's blood tests, but the antibodies can go back to normal after a long-term infection. So stool PCR testing, as done at GI map, is kind of a gold standard, accuracy-wise way to find it. And oftentimes you'll have patients who find it on a GI map and they'll miss it on other labs. That's very common. So, and again, most of the other lab techniques are looking for acute infections. We're worried about chronic. And this is just a note to remind myself, to tell you that, it's not just about H. pylori. You also want to do the dietary changes. Oftentimes you're going to have other pathogens involved. And so it's, a little more complicated than just getting rid of the one bug. Then you want to sequence it, right? And what's the sequence? Well, you want to address the, let me skip back here for a second. Here's the sequence. You want to test and correct the adrenals, get the lifestyle stuff figured out, get them on a good diet, give them a month or two of that, and then aggressively go after the H. pylori. So you want to treat the stress response, get that cleared up, 1 or 2 months, and then go after the H. pylori once you feel like they're kind of snapping out of that. It's hard to get rid of H. pylori in someone who's stressed out of their mind. Is another way of sort of summarizing all that. So H. pylori treatment, my medical doctor friends, I'm a chiropractor by training, so I can't prescribe stuff, right? But my medical doctor friends use Prev packs all the time. It's a lot of antibiotics, and they're hesitant to do that unless they have to. I think most of the MDs that I'm good friends with prefer to use the herbs if they can, but they use the medications if they have to. In my practice, I'll refer out if it's urgent and the person's bleeding and they have a big ulcer and they need a prescription. But 92% of the time, I'm going to treat with the herbs: Mastica, slippery elm, DGL. I also use zinc carnosine a lot in the programs. And of course, probiotics, multi-strain probiotics, high potency. Sometimes we'll even do 2 or 3 different types of probiotics simultaneously, like a bifidobacter, a spore-based one. Maybe sometimes, my favorite probably is Saccharomyces boulardii. So 2 or 3 different probiotics at the same time to really try to recolonize and get that working. And then of course you can do anti-inflammatory stuff like GI repair powders. Now, a special slide here for kids, because I've treated a lot of kids with H. pylori, and I don't know why, but it just seems to happen to children a lot, and it's really quite devastating. They'll have nausea, they'll be throwing up every day, they're not able to go to school, stomach pain every time they eat a piece of pizza, that kind of thing. And I had one summer, I don't know, when was this? This is like maybe 2005 or something like that, where I had a clinic in San Francisco at the time. In the back, and this is a true story, I started an organic yoga clothing line. Don't ask me why, because I don't know why. But anyways, I started an organic yoga clothing line. It was actually very successful for a short period of time before it completely failed. And in the back of the store in San Francisco, I had a clinic, which sounds crazy that I'm telling you the story. But anyways, that summer in San Francisco, I was treating patients, and these kids started to show up. Like every week, there'd be another kid with H. pylori. It was getting, by the end of the summer, I was just like overwhelmed with all these children. It's just like maybe 10 to 16 age range that all had H. pylori. They all had the same symptoms. It wasn't even complicated. They have everything on this page here. They all had stomach pain and nausea. They were all throwing up. They couldn't focus. They couldn't do well, in school or athletic activities, and they were tired. So, don't be shy about testing kids. It's an important thing to deal with, whether it's an adult or a child. And then it's an old patient of mine. I like to use as an example. Fatigue, insomnia, woke up at night with stomach pain. This was one of the first times that I realized that H. pylori could cause insomnia. And what happened with Jesse was we treated the H. pylori. He came back. He's like, I just realized I was waking up at night because my stomach hurt. I was like, really? He's like, yeah, I didn't even know that's what was happening because it stopped happening and I'm sleeping through the night now for the first time. So he didn't even put together that he was waking up because his stomach hurt until it stopped. So he didn't come in and say, oh, I can't sleep at night because my stomach hurts. He didn't know that was happening until it went away. It went away right when we treated his H. pylori. He also had severe amino acid depletion where transmitter levels were really low. So he had anxiety and depression and fatigue and the things that you would think about that would affect you cognitively, right? If you can't make enough brain chemicals. And he had blastocystis hominis, which is another infection that can show up. It's paired with H. pylori often. Why, I don't know, but those often are paired together. And all these problems started when he was under high stress playing on a college level basketball team, college sports, Division A or whatever they call it. I don't know, like one of the top teams in the country. Not top 10, but maybe top 20 or 30, something like that. And super high stress, working really hard, playing basketball in this college team, and whole life fell apart. Right? And so it's not always exactly obvious. And be sure you check kids. Be sure that you are checking people that don't have digestive symptoms. They're saying that they can't sleep at night, but they're not aware that it's because of the stomach pain. Integrative treatment programs. So number one, adrenal support. I like to do the salivary testing or the Dutch test and get those done, and then correct problems using DHEA and pregnenolone. Do a lot of teaching around adrenal programs. And that to me is like round one in reducing the stress on the HPA axis so that you can effectively knock out the bug with either medications or herbal products. I always prefer the herbs if you can do that. And then recolonize with the probiotics that we mentioned. Okay, so step one, again, stress and diet addressing. Step two, killing the bugs. And then step three, the repair process. Now, I'm going to say this one more time because I think this is what I see most people messing up, where they're trying to kill the H. pylori or the Giardia or crypto, whatever the bug is, before they test and correct the adrenal hormones. And this again is right out of Dr. Timmons' playbook. He wanted to prepare the body to treat the GI infections. He thought the GI infections were the most important thing, but he wanted to maximize the odds that the program was going to work, right? By tilting the playing field in his favor by working on the adrenals, strengthening secretory IgA, cleaning up the lifestyle stuff to make all that, more likely that it was going to work when he got to the killing point. Now, I just threw this in. I'm not going to read the slide. I hate it when people read slides. This is just a reminder for me to say, hey, wait a minute. H. pylori is not always bad. And you can read the slide later if you're really kind of curious about it. But the summary here is that H. pylori can be protective. There's research that shows that H. pylori can improve digestion or improve the immune system or improve the person's overall health. Those are not the people that are coming into my office when, and I'm sure there are millions of people out there in whom H. pylori is a positive force in their life. But in my practice, when we get rid of H. pylori, people get better. So I am a strong advocate of treating it if you find it. And I'm sure the people that have H. pylori for whom it's protective and it's a good organism probably just are not sick, right? They're not coming into offices like mine. So I have a very distorted view of this based on the fact that I treat people who are pretty sick. So key concepts to summarize, and then we'll look at some case studies. Number one, H. pylori causes damage to the stomach lining, reduces stomach acid production, so amino acids are not absorbed. Chronic inflammation and low amino acid levels impacts every cell in the body. I think that should be a singular, impact every cell in the body. All right, let's just think about that for a second. H. pylori can cause digestive symptoms. Everyone agrees on that. That's relatively easy. I don't think you need to attend a webinar for someone to tell you that. The point that's extra special here is H. pylori always causes damage to the stomach lining and reduces stomach acid production and interferes with amino acid absorption. Whether the person has stomach pain or not, whether the person has GERD or not, whether the person is vomiting or not, if they, even without any digestive problems, there's always going to be inflammation and a block of absorption of amino acids. If you don't believe me, Google H. pylori Nobel Prize Committee, because it is directly on that Nobel Prize Committee report that in all patients, H. pylori causes inflammation, right from the, scientific kind of oracle, right? Nobel Prize folks don't mess around, and they don't write things that are kind of silly. When I read that, I was like, whoa, guys, hey, that's naturopathic medicine. I don't know if you knew that, but anyways, it's not symptomatically oriented. So the second thing here, reduce inflammation through diet and lifestyle changes. That's the next big concept, right? You don't just kill. Gotta get the person ready. Strengthening secretory IgA levels. That's through adrenal programs. Okay, we'll talk about that some other day, but that's a whole other kind of thing you should learn how to do. And I use DHEA and pregnenolone for the adrenal programs. And then kill all the bugs. I always do H. pylori first, but you may do others later, depending on the case. And let's see here. Oh yeah, let's talk about this one case. And then I have these labs I can pull up to here. Let's look at this so you can see a lab. All right, so here's pretty much what a lab looks like. And we're just going to focus on this one infection. We could do this all day long, talking about other bugs. But here's your GI map, and here's H. pylori. Pretty straightforward, right? They say hi, it's in red, kind of hard to miss. Although, and I'm not making this up, I don't know if any of my students are on this call right now, but this morning in class, Dr. Steve, I think it was Steve. I don't know. I think it was Steve, either Steve or Jason. This morning in class, one of the doctors presented a GI map in class. It was run by a different practitioner. The H. pylori was positive, and the other practitioner was like, take some probiotics. And the doctor in the mentorship program was like, hey, Dan, I got this lab in. I'm not really sure. I've never seen one of these before. It was his first GI map. He's like, some other doctor ran it. But did they do the right thing? I was like, nah, I don't think so, Steve. I think you should treat the H. pylori. And this patient had a massive amount of digestive symptoms that have been going on for years. So anyways, I don't think this is that complicated, but some people miss it. Maybe they don't get the point. If H. pylori is positive, and they have anything to do with a GI symptom, or they have anything to do, you're suspecting now, right? Amino acid absorption problem, you're tired, depressed, something like that's going on, treat it. You're going to get better. I don't know why people would, anyways, we don't want to get into that, but that just happened this morning. Similarly, people can have dysbiotic bacteria. Again, I find this pretty easy to interpret. They're doing everything they can here for us, right? It's in red, that's bad. Red is bad, high is bad. Okay, so red is bad, high is bad. Dysbiosis and no other bugs, okay? So, oh, and here, the secretory IgA is low. So that's a poor immune response in the gut lining. The immunoglobulins are not doing that great. And they have a gluten-related problem. Okay, so this person needs an H. pylori treatment and maybe later a dysbiosis treatment to clear things out. And let me show you a typical patient presentation here. And these are like just my favorite H. pylori cases. I mean, I've had a couple thousand of these people, but this one I just remember so well because it was so crazy. So this woman, you know, otherwise kind of healthy, normal human person, working as a bartender, and she got diagnosed as having depression and an eating disorder. And she was referred to me by an eating disorders professional counselor who was like, Dan, I'd like you to run some labs on this woman. She's like growing up every day. And so basically she would wake up every morning and be nauseous and then start throwing up. And so she didn't want to eat because every time she ate, she would throw up. And so the eating disorders counselor was like, I don't know, Dan, she doesn't seem to have the normal eating disorders history. You know, she's not trying to lose weight. She didn't think it was a psychological problem. And it turned out this poor woman had H. pylori. And as soon as we got rid of the H. pylori, her stomach pain went away. She stopped throwing up all the time. And then her mood got better because she was absorbing amino acids finally. You know? I mean, I will never forget that. That was like a life lesson. It's like, whoa, you could really get a distorted view of things here if you're not careful. All right, let's look at another test here. And we're going to leave a lot of time for questions because I think there are a lot of questions that were coming in. So here's another case. Okay, again, it's in red. It says hi. If you see that and the person has what? Any GI symptoms or anything that you suspect is related to not being able to break down protein. What's that? Antibodies and a compromised immune system, hemoglobin and a fatigue problem because you can't carry oxygen that great, insulin-related problem because you can't make insulin unless you have all the amino acids. So they could have a blood sugar handling problem that H. pylori is triggering. Remember all those slides on proteins? Oh, pyruvate dehydrogenase complex or succinate dehydrogenase, those enzymes that help the conversion of glucose go to acetyl-CoA so we can make energy. So most people that have H. pylori are going to have one or more of these problems. They're not going to carry oxygen real well. They're not going to be able to make energy out of glucose really well. So the blood sugar is going to be a little whacked out because insulin is just not that great. Their neurotransmitters are going to be messed up, right? Those are the consequences of H. pylori. That's why it can affect the whole body because it's centered in the stomach where protein digestion begins. Okay, so a high H. pylori, this person has some low and high markers on the normal bacterial flora. That's probably for some other day. We can talk about microbiome stuff. I'm very interested in that. I just don't have time today. Okay. And then dysbiotic bacteria, again, high and red is bad. Treat it, you know, as aggressively as you feel comfortable with. And then on this particular one, zonulin elevated and secretory IgA low. So this should be pretty straightforward for you guys to read the labs. And again, the treatments then are going to be in anyone that you suspect has either of these things, either the GI symptoms that are obvious or the amino acid absorption problems that are perhaps, I would say more common, but not as I don't want to say subtle, but you just have to think through that stuff a little bit more, okay? So we're going to open it up for questions in a minute. I just want to mention that we, again, for those of you that came in late, we have a mentorship program starting now. If you're interested, you can get a discount on that. The mentorship program, we start a new one every couple months. If you're not ready now, you can do one a few months from now. It's a one-year program, kind of deep dive into all these different subjects and put together programs comprehensively for clinical treatment. That's a one-year class that we do. And then we have a boot camp coming up.
So we enjoyed the video. We'll go to the presentation. Hopefully it is clear for you now. I think there is one question about what herbs are proven to be effective in H. pylori eradication. Actually, this question is out of the scope of the lecture because we don't deal with herbs. But anyhow, olive leaves extract have been proven to be effective against H. pylori eradication. H. pylori, a corkscrew-shaped, gram-negative rods reside in the mucus layer of stomach, human stomach. Unrecognized until 1980s, the organism is extraordinary common, infecting at least half of the world's population. H. pylori infection invariably caused acute and chronic gastric inflammation, but in only minority of cases does it result in overt clinical disease. Yet, the disease attributed to H. pylori infection, peptic ulcer disease, and gastric malignancy are among the world's most important causes of morbidity and mortality in adults. For this reason, the past 30 years have borne witness to various activity concerning diagnosis, treatment, prevention of this recently rediscovered pathogens. H. pylori does not invade beyond the gastric mucosa and does not directly cause fatalities. Mortality statistics for peptic ulcer disease. The data reveal decreasing mortality rates from peptic ulcer disease over time. Highest mortality rate has been observed in person born at time of industrial revolution. H. pylori is not a reportable disease. Survey are the most widely used method for studying the population distribution of H. pylori infection. Household institution of population survey have been conducted in numerous populations worldwide. H. pylori is a relatively fastidious organism that does not thrive outside its natural environment. Despite this, the organism can culture from gastric mucosa if proper measures are taken. H. pylori grows slowly and plates may require 7 days incubation to develop recognizable colonies. Histology is still considered by many to be the gold standard for diagnosis. Another commonly used endoscopic test is the rapid Urase test. This method exploits the organism's rapid hydrolysis of urea to ammonia and carbon dioxide. 3 non-invasive methods to diagnose H. pylori are widely used: serology, breath test, and stool enzymatic assay, HEA. Numerous commercial serological tests, including Anopheles rapid test. At their best, adult and older children ELISA kits have sensitivity and specificity of almost 90 and 95%. These tests, which target the 128 kilodalton protein of the CAG-A pathogenicity islands, have become important in epidemiological survey of population at risk. The genus Ulobacter, recognized with this rediscovery of H. pylori, comprise a diverse group of curved spiral bacteria that colonize the gastric and intestinal tracts of animals and well as humans. Expression of adherence factors may be adaptive, with organism able to exploit both phase variation. Accounting for 50% of proteins by weight, this enzyme, nickel-contaminating hexamer, hydrolyze urea to CO2 and ammonia. Crystallography has revealed a large 1.1 megadalton supramolecular assembly consisting of 12 catalytic units active to a pH of 3. Commonly thought to be constitutively expressed, recent evidence suggests that urease activity may be more controlled. Because acute infection invariably pass undetected, acute H. pylori infection has rarely been diagnosed outside experimental or iatrogenic settings. In the absence of specific antimicrobial therapy diagnosed to eradicate the organism, infection with H. pylori typically remains in the stomach for decades. In older adults, however, H. pylori paralyzed and serological response may diminish. Despite histological evidence of inflammation, the majority of individuals infected with H. pylori are asymptomatic. What is known about acute symptoms of H. pylori comes largely from experimental infections. In his self-inoculation experiment, Barry Marshall was asymptomatic until day 7 when he developed epigastric discomfort, vomiting, and irritability. Among 20 subjects given H. pylori after acid suppression, 18 developed infection. All of these developed mild to moderate gastrointestinal symptoms. With naturally acquired infection, much less is known about symptoms. In a study of 175 persons. Non-ulcer dyspepsia, the role of H. pylori infection in non-ulcer dyspepsia remains controversial. Exploring the relationship is problematic because of the wide variety of symptoms and fall under this catch-all term. The same inconsistency may reflect the lack of the control group. H. pylori is curable with antimicrobial therapy. The two most widely used regimens are 10 days of proton pump inhibitors triple therapy or 2 weeks of bismuth triple therapy. Whereas consensus has been achieved on antimicrobial treatment, controversy raged over whom to treat. Still, others maintain the eradication of H. pylori in all those infected, regardless of symptoms. No vaccine is currently available to prevent or to treat H. pylori infection, although vaccine development is an area of considerable interest. Since the mechanism of transmission of H. pylori is still uncertain, there are no current recommendations for control and prevention. Since 1982, enormous amounts of research has been conducted on H. pylori infection, and the last 5 years alone, over 6,000 manuscripts were published. Neither symptoms of naturally acquired acute infection nor transmission route are fairly established. Over half of the world population is infected with H. pylori. Developing countries, H. pylori is still almost universal. In developed countries, on the other hand, the incidence of infection has been rapidly decreasing without any specific antibacterial intervention. Should we be focusing on the vaccine development for a disease that disappears with improvement in hygiene? Should we be screening and treating persons in order to prevent H. pylori-related disease? Does H. pylori have any benefits? Would widespread use of antibiotics to treat H. pylori produce more harm in terms of overall burden of antimicrobial resistance than good? These very fundamental issues remain to be resolved. Any question يا اخوان? We'll move to another video. فإذا في أسئلة الآن before we move to the video, next video. حسام واضحة, thank you very much. ريان, thank you. زينب, دانا, هيام, سمير, مشعل, عبد الله, سماح. Okay, we'll move to a video presentation, أخوان. Let's again discuss how to handle an H. pylori.
So, H Pylori, also known as Campylobacter pylori.
H pylori is an infection that's actually has a fair few consequences of it and is associated with chronic gastritis, peptic ulcer disease, gastric adenocarcinoma, and gastric mucosa associated lymphocytic lymphoma, also known as MALT lymphoma. And interestingly enough, H pylori was first kind of ignored in the old days.
Apart from recall, I will give you the link. I've also certain doctors in Perth
actually drank liquid with H pylori. Those doctors were Dr. Barry Marshall and Dr. Robin Warren, and it actually showed that the H pylori itself caused peptic ulcers, kind of challenging the belief at the time that they thought you get ulcers in the stomach mainly from stress, spicy foods, and too much acid. And they received a Nobel Prize for this in 2005. So a little bit more about H pylori. It's the most common chronic bacterial infection found in humans to date. And most people actually get it at a younger age. And there's been multiple studies done that actually show most people that have H pylori get infected before the age of 5 years. The reason why this happens and the mechanism behind this is actually quite debated at present, and it's still not really proven how it is. But the main transmission is believed to be by the fecal oral or oral routes. However, this hasn't been proven. Once someone gets infected with H pylori, they might actually have it for many decades, and they may or may not get any symptoms, and some of them can have it lifelong without any ulcers forming. Most commonly, H pylori seems to be associated with individuals from African American descent, Hispanic descent, and Native Alaskans, compared to Caucasian population. The risk of getting H pylori seems to be strongly correlated with someone's socioeconomic status and living conditions at a very young age. So people living in very close proximities and being in overcrowding, the amount of siblings you have, and if you're sharing a bed with someone else, and difficulties with clean, fresh water, all these factors have been associated with an increased risk of H pylori infection. The way they kind of figured this out and got evidence behind this was with Japan. So in Japan, up to 80% of the adult population that were born before 1950, so around World War II, were infected with H pylori. However, only 45% of the individuals born between 1950 and 1960, and only 25% of those born between 1960 and 1970 were shown to be infected. So it showed a rapid decline of infection rates in individuals, which was attributed to Japan's economic progress and improvement in sanitary after the World War II. H pylori can cause reinfection. However, it's quite unlikely, and current data shows that H pylori following a successful cure with antibiotics is very unusual, and the current rate of that is less than 2% of persons per year, which is a similar rate to people actually getting the initial infection. So basically, once you have treated, the chance of it coming back are the same as someone that has never had it before. So what do I find interesting about H pylori? I mean, it makes it such an interesting bacteria is a little bit about the properties of H. pylori. So it's spiral shaped. It is microaerophilic. It is gram-negative. It produces urase. Lots of subspecies produce catalase, and it has specific adhesion receptors. And on the higher microscope, it actually shows it has 2 to 7 sheeted flagella that help it move. Furthermore, H. pylori is able to form cocoid forms outside the body and being able to survive for many, many years in both feces and water, especially stagnant water. So what makes H. pylori so ideal to live in the stomach under the harsh conditions that exist there? So probably most important is the urase that it produces. The urase itself hydrolyzes urea that's in the gut to form ammonia, which helps neutralize gastric acid and protects the H. pylori bug from the acid around it and kind of allow it to penetrate up to the gastric mucus layer. And bacterial urase activity is clinically important because it allows us to do certain testing, such as a urea PrEP test. The shape of the bug, given its spiral shape with a flagella, it allows passage through the mucus layer quite easily to the gastric surface epithelium. And on that epithelium, there's certain specific receptors that the bug adheres to. Now, these receptors are different between individuals and strains, which explains why some people are more prone to H. pylori, certain H. pylori strains compared to others. And then the catalase it produces is to help neutralize the damage by reactive oxygen metabolites, which neutrophils nearby liberate. Furthermore, of important note is that H. pylori doesn't actually get into the gastroduodenal tissue. Instead, what it does, it creates kind of an environment that allows the acid in the stomach to penetrate and hit the underlying mucosa, causing acid peptic damage, disrupting the mucus layer. And the host immune response, so the body then trying to clear the H. pylori, creates a cascade of further inflammation, leading to further injury. This chronic inflammation caused by the H. pylori upsets the gastric acid secretion in different stages. And this kind of leads to the chronic inflammation of the gastric lining or gastritis. Now, most people might get a small ache, but kind of ignore it, and it doesn't seem to progress, and it kind of stays where it is. However, in some individuals, this altered secretion and including an ongoing tissue injury leads to a peptic ulcer forming. While in some other cases, the ongoing inflammation leads to atrophy and then metaplasia of the intestinal mucosa, which can lead to gastric carcinoma, or in some cases, the persistent immune stimulation of this can lead to that mild lymphoma. And I have this slide coming up. Here we go. It kind of explains that. So you have, if you look at the first points, you have the gastric fluids, and then you have the mucus layer, which the H. pylori goes through, then sits at the epithelial cell, where it produces lots of urase, creating a nice and neutral environment. And then it replicates, as you can see, and it creates this nice environment that basically eventually allow the gastric fluid from this part here to basically invade, cause inflammation and damage to the mucus. And then you get all these leukocytes involved as well and increase the risk of that mild lymphoma. And I think this is quite a nice picture to explain that all. Now, apart from the treatments we know, there's actually done some studies trying to do vaccination and seeing if we can vaccinate people against H. pylori. And there was a randomized phase 3 trial recently done that used almost 4,500 H. pylori uninfected children, so people that did not have H. pylori, between the ages of 6 to 15 years. And they got a 3-dose oral H. pylori vaccine or placebo in a 1-to-1 ratio. And it actually showed the vaccine to be efficient in up to 72% of cases. Among the patients who completed the extended follow-up over many, many years, the H. pylori continued to be lower in the vaccinated compared to the unvaccinated children. However, protection levels were lower in the second and third year after the vaccine, and it showed that the vaccine actually reduces every year with efficiency. In regards to side effects, there were no serious adverse events related to the vaccine, and there was only a mild increase of nausea and vomiting in the vaccine arm compared to the placebo arm. Unfortunately, this is still going for long-term follow-up to see it's feasible to create quite expensive vaccine for this condition. So the most important part is you have a patient, you're concerned that they might have H. pylori. When do you actually test for it? So first and foremost, you only test if you're actually planning on treating and whether you're planning on treating this patient. So if you have a patient that even if they're positive is not going to get treatment, then it's of course redundant to do this. So I've kind of tried to color code when you check for H. pylori. So in the most important case, anyone that has peptic ulcer disease or a history of peptic ulcer disease in the past, and there has been no clear documentation whether H. pylori has been cured or has been cured in the past, they need to get a H. pylori test. Furthermore, anyone with multiple lymphoma diagnosis needs a H. pylori test and urgent treatment for that. To treat the H. pylori to prevent worsening of the lymphoma. Furthermore, anyone with gastric cancer needs a H. pylori test. The one in the yellow or orange here is a little bit more complex, and there's no evidence either way whether it's beneficial or not. So anyone with dyspepsia that is less than 60 years old can be considered for H. pylori tested. Of course, anyone over 60 should get an upper endoscopy. Any commencement of long-term NSAIDs or low-dose aspirin. Here in Australia, we don't routinely test for H. pylori for someone that has been on NSAIDs or low-dose aspirin for a prolonged period of time. There's actually no clear evidence whether it's beneficial or not. And currently, it's kind of clinician dependent. The same goes for iron deficiency anemia. And there's actually a strong link that individuals that have idiopathic thrombocytopenic bacteria are quite often H. pylori positive as well. But it's a little bit of a chicken and egg, which ones came first, and there's currently no evidence of that. There is no evidence whatsoever that it's beneficial to test people for H. pylori if they have any family members, whether it's immediate or distant family members with gastric cancer, anyone with lymphocytic gastritis, hyperplastic gastric polyps, or hyperemesis brevidarum. Furthermore, what tests can we actually do to check for H. pylori? So there's multiple tests that can be done, but realistically, there should only be 3 tests that should be done. So the 2 non-invasive ones are the urea bread test or the stool antigen test. And then invasive-wise, we can test via endoscopy. So those are your only 3 options. Do not use any of the other tests if they do not show benefit. Furthermore, as in red, you can see below here, do not do any of these tests if this patient has been on PPI in the last 2 weeks or any antibiotics, no matter which one, in the last 4 weeks. So if you treat H. pylori, you actually got to wait 4 weeks after you finish the antibiotics before you test again for H. pylori eradication. And it's actually really important that all patients treated for H. pylori should have confirmation of eradication because of the increasing antibiotic resistance in the community. So, in regards to treatments, first and foremost, any patient that has an H. pylori infection should be treated. Now, the complex part is there's so many options to treat it. Which one do you actually use? Especially given increased macrolide resistance, and a lot of patients in Australia, and I'm sure with you guys as well, have a penicillin allergy. So there's some things to think about before you consider treatment is what allergies do they have, and is it an actual true allergy, or is it a little bit of nausea when they were a child? You can kind of use your clinical guidance on that one. And furthermore, are there any histories of any macrolide resistance in this patient, which kind of makes you steer away from medications. Now, these are the guidelines on the left side that are recommended in Australia. These are the guidelines on the right side that are recommended by UpToDate. But the one in Australia we use on the left side is the first line therapy is by using esomeprazole twice a day for 7 days, amoxicillin 1 gram twice a day for 7 days, and then clarithromycin twice a day for 7 days. Now, unfortunately, these are quite old guidelines in Australia, and they have not been updated in the last 15 years. And all the studies that kind of came up with this consensus and provided this evidence are all from prior to 2010. So it's not very up-to-date medication. In Australia, especially, we're getting increased rates of levofloxacin resistance. So we're trying to avoid it in Australia to use unless the H pylori strain has been cultured and is known to be sensitive to levofloxacin. However, of note of that, it is noted that levofloxacin resistance is very prevalent in Europe, Australia, and the United States. But I've looked at some data on PubMed, and there seems to be very low rates of levofloxacin resistant with H pylori in Southeast Asia and in South America. So I think for you guys, it is something to consider levofloxacin, and we'll get to that in a second. In regards to first-line therapy here with clarithromycin, I'm a big fan of not using clarithromycin, and I'll explain a little bit further why I think that. There's been many studies done in Australia and other countries of increasing rate of clarithromycin resistance, H pylori. And in Europe, East Mediterranean and Western regions, the rates of primary clarithromycin resistance was sometimes over 50%. So that's 50%. So it means half of the H pylori was basically resistant to clarithromycin. And some studies done in Australia actually recently on patients with known H pylori via endoscopy. So this is, you've got to kind of think about it. You have a little bit of a biased population because these were patients that need an endoscopy for multiple indications. You do got to think about it. They showed up to 90% resistant clarithromycin. And I'm not sure what the resistant rates are in Burma, but over the whole world, there seems to be more and more increased rate of clarithromycin. And in vitro, in the lab, exposure of clarithromycin, even for quite a short time to H pylori, seems to cause a lot of resistant increases. So there's kind of a little bit of background thinking that maybe this initial therapy we recommended worked for quite a long time purely because of the amoxicillin and the esomeprazole there rather than the clarithromycin. If you look at the UpToDate guidelines, which are mainly based on the United States, the way they look at H pylori is to basically at any prior macrolides in the past, so if they had clarithromycin for anything in the past, or if the clarithromycin resistant in the local area are greater than 50%, you kind of follow that as a guideline. And if there is any reason to think there might be macrolide resistance, they jump straight to quadruple therapy. I'll go to these Australian guidelines for the next step. All right. So this talks a little bit about salvage therapy in Australian guidelines, but I kind of use it here as a guideline for us to show what other types of therapy are out there. And this is probably the main one I would recommend at the moment in Australia and potentially in Burma as well, given you don't get to use the clarithromycin. Levofloxacin is a little bit more difficult in Australia to get, but most hospitals can get access to it. And then amoxicillin is readily available and has very good activity against H pylori. And we always need a proton pump inhibitor. Now, of note, this is the guideline from Australia if the previous therapy on this page has failed. You try this as a second line. Now, what you should know is that it says for 10 days, or the previous one says 7 days. And I'll jump into this in a second. There's actually a moderate evidence-based guideline, so it's not very strong, but it actually suggested now that we aim more for 14 days rather than 7 or 10, especially if the patient can tolerate this. Now, ideally, I personally recommend this one if you have access to levofloxacin, or otherwise use the quadruple therapy here below as one of the guidelines Australia recommends. But I would be considerate of the metronidazole at the bottom and potentially change that for amoxicillin. And I'll explain that in a little bit. But basically, amoxicillin and tetracycline have the best activity against H pylori. And bismuth is fantastic if you can get your hands on it. However, I don't know if any of you have ever tried it. It is quite unpleasant to take for the patients, and we'll have to consider whether they're going to be adherent or not, especially given that they take 4 times a day. So you really got to pick your patient. If you have a highly intelligent, motivated patient that you think will stick to, a tablet of proton pump inhibitors, then, up to, depending what strength bismuth comes in, 80.60 or 120 here, you're talking about another potentially 8 tablets, then another 4 tablets, and then another 3 or 4 tablets, depending if you use metronidazole or amoxicillin. You're talking about, quite a large amount of tablets, especially in young people. You have to consider whether they're actually going to be adherent or not. And then finally, we try and use this in Australia basically as the last line therapy, because the rifabutin is kind of our last line of defense in Australia for a lot of infections. And what we aim to do is basically only leave that for people as a last line defense, given the increasing rates of resistance. So I think someone is drawing on the screen. I don't know if they're trying to ask something.
Screen ပေါ်မှာလေdrawing လုပ်ရတာ Dr. Loudon. Sorry, Shores, I think someone might be doing it.
No, that's all right. We'll just get to it. Sure. As long as they're right.
Sorry.
Yeah.
No, don't stress. All right.
ဆရာလောက်ဟို screen ပေါ်မှာ drawing လုပ်ထားပြီးချက်ပြီးပါဦးဆရာ
We'll go to the next page anyways. I think we can still see it. Should be okay. So I've just taken you guys through the Australian guidelines. This is the American guidelines. If someone has tried, triple therapy with clarithromycin base with clarithromycin in it, or if they've tried bismuth quadruple therapy, and you're trying to think, okay, that didn't work. We waited 4 weeks after their last antibiotics. They've not had a PPI for 2 weeks. And then you test them again, and the test comes back that they still have H pylori. Kind of follow this guideline, this path now. So if they've had clarithromycin-based triple therapy, and they're allergic to penicillin, you can try the bismuth quadruple therapy. Personally, that doesn't make any sense, because if someone had a penicillin allergy, they should have never been on the clarithromycin-based triple therapy. These are the up-to-date guidelines. I didn't write them. If they don't have a triple therapy, they kind of leave it in your hands. And they talk about this high-dose dual therapy, which is basically amoxicillin only at high doses. And sometimes people use up to 2 grams 4 times a day with a PPI twice a day. Or just levofloxacin, which we spoke about before, or the bismuth. Now, if someone tried the bismuth quadruple therapy, you can then either treat it with, again, bismuth quadruple therapy, with basically double the strength of PPI, or you can switch to levofloxacin triple therapy. And these are the American guidelines. I'm a big fan that if you've tried one therapy, the bismuth quadruple therapy, I wouldn't repeat it by just increasing the dosage of PPI. You're kind of trying to treat the same thing over and over again the same way, expecting a different result, which I personally don't believe. It didn't work once, it won't work the second time, so we just try something different. So I personally then recommend we head towards the levofloxacin triple therapy. And if they do have a Pylori FG, goes basically down the same ones we spoke before. And all of these therapies are explained at the bottom. All right, so I kind of spoke about the clarithromycin and its resistant levels and how we shouldn't use it. But this is a little bit of research done in Australia with our local pathology center in Australia. We have multiple. This is one of them. And they went through all the H Pylori testing they did between 2012 and 2018. Now, we've got to remember that a few of these samples were taken endoscopically, and there was generally some sort of indication to have that endoscopy done. So not all of these were primary H Pylori infections. So some of these might have been on H Pylori for quite some time. Had H Pylori for quite some time. They might have tried multiple therapies, and none of them worked. And then they kind of cultured it and looked for resistant levels. So this data is a little bit biased, but I think what's good about this data is this basically goes over a prolonged period of H Pylori testing. It shows what the resistance is to H Pylori. Most importantly, it shows that amoxicillin and tetracycline have very low resistant levels. And they quite commonly use medications. Most people have quite easy access to them. So sometimes a good first line therapy even is just plain amoxicillin and tetracycline with a PPI for 2 weeks and to see how that goes. Now, this data here, if you look at the levofloxacin, it's last time in 2018. And unfortunately, here in Queensland where we live, there's actually increased levofloxacin resistance now, which is quite strange given how difficult it is to get our hands on levofloxacin. Only really in hospital we can get it. And as you can see, rifampicin quite quickly builds resistance to it despite the little use. And as previously mentioned, there's very high levels of clarithromycin and metronidazole resistance. Interestingly, there's been a few studies done, and even if H. pylori is resistant to metronidazole, given metronidazole to these patients, even though they have metronidazole resistance, she'll show clearance rates that are very similar to patients treated on, for example, amoxicillin and tetracycline susceptibility. And they can't fully explain it, but I think it might be some of the other effects metronidazole has that aren't antibiotic. But I think they're still doing some research on this. So this is quite a busy slide, but I'm going to try and summarize where the difficulties are in H. pylori and some strong statements of what we should consider. So there's a group of European doctors, and they hold this conference every 3 years, either in the Netherlands or in Italy, in Florence. And as you can see, it's called the Maastricht, which is a town in the Netherlands. V, because it's the 5th conference they have, Florence Consensus, and then Florence is a town in Italy, and they just call that because they alternate where it is. This is published in 2016 and was meant to be revised in 2020. Fortunately, COVID kind of stopped all these doctors coming together, and they tried to, not mingle and mix. So they put it on hold now, but there's plans of having this conference again, or this workshop this year again. And what basically happens is there's a few infectious disease specialists, a few gastroenterologists, other doctors with an interest in H. pylori that come together, and they go through all the research published in the last X amount of years. So they have it now in the last 6 years, and they form different working groups over the days, and they go through all the evidence, and then they go through their clinical expertise and what they have seen in practice, and kind of show what their recommendations are, and they kind of make a couple statements as I've shown here, which I think are important ones. But if you have time, I recommend you have a bit of a read through this paper. You can find it online. But basically, what one of their statements is, if there's clarithromycin resistance less than 15%, you can consider using clarithromycin-based therapy. They recommend you can do antimicrobial susceptibility testing after the first treatment fails. However, the level of evidence is quite weak, but they strongly recommend it. And it's also partially to keep data in your local community of resistance. They go into this new guideline or recommendation of, as I showed previously, even the guidelines in Australia still recommend 7 or 10 days of treatment, but I actually strongly recommend we extend this to 14 days. Even though for most antibiotic treatments, we try and reduce the amount of therapy people get to try and reduce resistance. There's a strong recommendation with moderate level of evidence to actually give 14 days of PPI plus antibiotic therapy for H Pylori treatments. Their statement is that currently we give PPI, amoxicillin, clarithromycin, and, for example, metronidazole together.
Hello, Osho. Sorry for interruptions.
Okay, I think that's it for H Pylori. Any question, Akhwan, before we go to the break of Maghrib prayer? Any question, Akhwan? There is a question by Dr. Abdul Salam. He's saying, I didn't get when do we have to use salvage therapy? Can we please elaborate on it? The question here is really, thank you very much, Abdul Salam. This is an important question because some of the patients, they cannot have a good response to H Pylori treatment. And as we explained in the video and the lecture, there are many resistant in some antibiotics, and this explains the recurrence of the infection. So using salvage therapy are still not agreed upon universally, and most of them are still in the trial stage. There is no final conclusion. But bismuth more utilization is one of the cornerstone of the salvage therapy for H pylori. Any question يا اخوان regarding H pylori? Thank you هالة, thank you عبد السلام. تحضير امس يا ماجد صراحه ما اعرف عنه, يعني لازم الادمن هم يشوفوه, يعني فما ما اعرف والله, بس هم حيشوفوا رسالتك ما عليك. يعني. وايل, ماجد, thank you very much. فهد, افراح, واضحه. كارن, thank you very much. نوال, انفال, thank you. عبد الله, منى. في واحد يا اخوان اسمه زوم يوزر هنا, فبس يا ليت ينتبه اللي يعدل الاسم, يتاكد بس انه نفس اللي مسجل, يعني ما يكون نيك نيم وما نعرف ايش معنى. ريان, ساره, سماح, ايام, ثانك يو فيري ماتش. امل, مشعل, عبير, محمد, نوره, سماح, فارس, عبد الرحمن. والله اتوقع ان شاء الله بيردوا عليك الادب, اسمك موجود عندهم. سلمان, عبد الرحمن, رقيه, هاله, عبد السلام, ثانك يو فيري ماتش all of you. طيب يا اخوان, محمد, بس ما هو, لحظه, محمد الشرقاوي, ايوه خلاص, نعم. انت تلاقي اسمك, افتح البارتيسيبنتس, حتلاقي اسمك موجود, سواء نيك نيم او الاسم, تقدر تعدل الاسم طبعا. نجاة, هاني, ساره, محمد, ثانك يو فيري ماتش all of you. طيب يا اخوان, نلتقي بعد الصلاه. نعيمه, طبعا ما شاء الله 600 وشويه, صعب يا اخوان ترى اجيب الاسماء كلها. نعيمه عبد ما ادري. طيب يا اخوان, we'll be back بعد صلاه المغرب ان شاء الله.
Welcome back, everybody. Assalamu alaikum. We will resume our presentation. Our next topic is about legionellosis. Legionellosis is an infection with bacteria of the genus Legionella. It is primarily associated with two clinical and epidemiological distinct syndrome, Legionella disease, but potentially fatal form of pneumonia, and bronchial fever, a self-limited non-pneumatic illness. At least 49 Legionella species and 71 serotypes have been identified, but one species, Legionella pneumophilia, causes over 90% of Legionnaire disease cases. Although Legionnaire disease outbreak capture much media attention, the majority of reported cases are sporadic. Less than 5% of cases are reported in public health authorities. Bronchial fever is generally recognized only when cluster of illness occur. Legionnaire disease is an important cause of community-acquired and nosocomial pneumonia. Direct immunofluorescent antibody staining has been widely used to identify the organism in lung tissue postmortem. Monoclonal DFA reagents are designed to detect only single species or serogroup polyclonal reagents, but are more likely to cross-react with bacteria that may colonize the respiratory tract, such as Pseudomonas species. معلش يا اخوان انا اوقف شوي احيانا اقرا الرسائل واقرا الكويستشنز، فيعني المعذره اذا وقفت الصوت. طبعا معلش يعني انا اوقف شوي احيانا اما اقرا رسائل ولا اقرا تجيني مسج من المنظمين، هذا هو سبب انقطاع الصوت. رابط التحضير ارسلناه يا محمد عسيري في الشات بوكس. The exact role of legionellosis in causing death may be difficult to determine because other serious life-threatening underlying illnesses are often present. Additionally, bacteria other than Legionella species occasionally may be isolated from the lungs or persons who have died of Legionnaire disease. Comprehensive, systematic, reliable data on mortality due to Legionnaire disease are unavailable. Although the National Center of Health Statistics receive copies of death certificates to tabulate national figure on death attributed to pneumonia. Data regarding the prevalence of Legionnaire disease among patients who have died with pneumonia have been systematically sought in one multi-city survey. Data regarding the prevalence of Legionnaire disease among patients who have died with pneumonia have been systematically sought in one multi-city survey of fatal nosocomial infection. Estimate of disease incidence from these studies are limited by small sample size and a non-random selection of hospital survey. Review autopsy material during epidemic investigation has been useful defining the magnitude of individual outbreak. The case fatality rate can be estimated by determining the proportion of fatal cases among those reported, although surveillance of Legionnaire disease. However, these estimates may be inaccurate because of unreported of cases. The limited data we have on the incidence of prevalence of Legionnaire disease in the general population are based on findings from studies of patients with community-acquired pneumonia. The most dependable data are from studies that are prospective and population-based. The findings of these studies are affected by the criteria used to identify patients for laboratory testing. Studies of incidence of and prevalence of healthcare-associated legionellosis are influenced by the same factor that affect studies of patients with community-acquired infection. Additionally, the incidence of healthcare-associated infection varies greatly from one institution to another because of the duration in hospital facilities and patients. Surveillance of legionellosis is conducted with varying intensity in many countries. Reports through NNSDFS from state health departments include only basic demographic information for cases of legionellosis. A supplementary paper-based surveillance system of legionellosis disease collects more detailed information, including whether cases are travel, healthcare, and/or outbreak related. However, data from the prospective studies of patients with pneumonia indicate that less than 5% of legionellosis cases are reported to CDC. In addition to modifying the case, in Europe, over 30 countries participate in European surveillance scheme for travel-associated legionella disease. In addition to timely reporting to European Working Group of Legionella Infection of travel-related cases. The indirect immunofluorescent antibody, IFA test, has been used most widely in serological surveys, although other less standardized methods, including hemagglutinin, indirect hemagglutination, immune adherence, and others. Differences in methods of preparing test antigen and possible cross reactions with other organisms. Serological tests prepared from heat-killed antigen, the most widely used test in North America. Rates of elevated IFA titers to Legionella pneumophilia. The relatively high background rates of elevated titers indicate that Western predictive value of single titer for determining recent legionellosis is low. Although serological tests are widely used for diagnosis of legionellosis, these tests have a number of important and often under-recognized limitations. Testing for IgM alone has not proven useful for the diagnosis of legionellosis. Positive IgM often increase concomitantly with IgG and may remain elevated for months or years after infection. A single elevated antibody titer doesn't confirm the diagnosis of legionellosis. And the largest study of patients hospitalized with community-acquired pneumonia in Ohio, single titer didn't distinguish between legionellosis disease and pneumonia. The sensitivity of DFA testing of respiratory secretions for the diagnosis of legionellosis disease has ranged from 25 to 75% and specificity more than 95%. The surveillance of legionellosis doesn't yield the accurate measures of incidence because many cases are not diagnosed or reported. Furthermore, only few population-based studies have assessed the incidence of legionellosis. After projection to the age, race-specific incidence of US population, the study estimated 8,000 up to 18,000. Cases may be diagnosed but not reported to the state public health department or from state of CDC. And study of patients participating in prepaid health plan that covered 10% over the population of Seattle. A much lower incidence was observed in one-year community-based study in Nottingham, England. A more recent prospective study in Barcelona, Spain, showed similar results. Although 90% of legionellosis cases reported through surveillance in the United States, risk factors of legionellosis have been identified primarily from epidemiological investigation conducted in outbreak setting. An outbreak of legionellosis, the risk of illness is often associated with the amount of time spent in the area after exposure. Although the organism is present in respiratory secretions of patients with legionnaire disease. Legionellosis occur worldwide, although most surveillance data are reported by countries in temperate regions. In the United States, the incidence of reported legionellosis disease cases varies greatly among the states. Rates of reported legionellosis disease cases vary seasonally, with the highest rates occurring during the summer months. Outbreak of legionnaire disease associated with cooling towers often have occurred during the summer and fall months. Healthcare-associated infections. Hospitals provide unique settings of the increased transmission of legionnaire disease because complex plumbing systems favor amplification of Legionella. Numerous outbreaks of healthcare-associated disease have been reported, and exposures of aerosol generated by cooling towers, showers, and faucets. In one hospital in Arizona, after 3 possible nosocomial cases were identified in 3 months period, increased retrospective and prospective surveillance. The risk of acquired legionella disease during hospitalization is influenced by a number of factors. A history of recent prevalence is associated with approximately 21% of legionella disease. Outbreaks of legionella disease have been identified among guests at hotel and resort and among passengers on cruise ships. Although socioeconomic status may be linked with risk factors for legionellosis, a direct association between socioeconomic status and the risk of legionellosis has not been identified. Inhalation of Legionella is aerolized droplets and nuclei is the primary means of transmission of legionellosis. The aerolized droplets must be a respirable size of 1 up to 5 nanometer. A number of devices have been implicated as a source of aerosol transmission of Legionella. The rule of thumb humidifier in sporadic cases of Legionella disease is unknown. Ducts and vents of air conditioning system can be a conduit for passages of aerosol containing Legionella organisms from a contaminated cooling towers. ايوه نفسه هذا حق اليوم الثاني، هذا التحضير رابط التحضير اليوم الثاني اليوم يعني. Legionellosis result from aspiration of upper respiratory secretions or gastric contents may occur among certain hospitalized patients. Erythromycin common prophylaxis of immunosuppressed patients at a hospital with high incidence of nosocomial Legionella disease due to colonization of the hot water distribution system resulted in protection against Legionella disease. Although animal models with vaccines of Legionella have shown some success, the development of vaccine for human against Legionella has not been attempted. The clinical presentation of Legionellosis disease, Legionnaire disease, is similar to that of other types of pneumonia. Therefore, accurate rapid diagnostic tests are essential to ensure appropriate clinical management of patients and early recognition of outbreaks so that prevention of strategies may be attributed. Although environments capable of supporting the growth of Legionella, such as cooling towers and hot water distribution system, are daily parts of life, and the bacterium is frequently present in these systems. More information is needed about specific virulence, host, and environmental factors. Prevention of Legionellosis is challenging because of the steps necessary to identify an individual source of transmission. Because of diversity of settings in which the disease occurs, because of the difficulty in eradicating Legionella from aquatic environments. I think that was the last slide. Any question, Yafwan? احنا اليوم ان شاء الله نخلص على بين 9 و9 ونص ان شاء الله. Any question يا اخوان؟ شكرا هاني، جودة، عبد السلام، سارة، عادل، فتون، طارق، عيسى، تركي، نورة، جيا، نوال، زايد، فارس، علي، دانا، وضحى، سماح، بدر، عبد الرحمن، آية، سارة، ريان، أحمد، سوزان، مشعل، في، حنين، زهرة، عبد الكريم، نجلة. إيه ممكن يا أخي، ممكن تطلع وتدخل مرة ثانية بالزوم. التحضير مرة واحدة. فاطمة، ابتسام، مرة واحدة في اليوم. دكتور غدير، دكتور فاطمة، عبد الرحمن، أفراح. والله دكتور عبد السلام احتمال شوية نقصر وقت صلاة العشاء عشان نخلص بدري، لأنه باقي 3 محاضرات، باقي الآن واحدة وباقي غيرها 2. اليوم أقصر من أمس يعني، فبنحاول نختصر وقت صلاة العشاء عشان نخلص على بين 9 9 وربع يعني بالكثير. نوف، نعيمة، وسيمة، عبد السلام، Eli, thank you very much all of you. طيب يا اخوان, to save time we'll move to the next presentation. It is about a Mycoplasma pneumonia. Mycoplasma are a class of less bacteria that cause disease in human, animals, and plants. They include a species recognized as among the smallest known free living cells. These microbes have very small genome and unique genetic features. Their temporary classification as viruses and fastidious growth requirements set the stage early for the use of diagnostic techniques better suited to viral detection. Systematic infection are well known in both immunocompetent and immunocompromised patients, but the host and bacterial factors that allow invasion from epithelial colonization sites are poorly understood. The focus of this is to respiratory and extrapermeability is caused by pneumonia, by Mycoplasma. The clinical presentation of Mycoplasma pneumonia respiratory disease is similar to that seen with other atypical bacterial pathogens such as chlamydia, pneumonia, and Legionella, and in some key infection, Staphylococcus pneumonia. Mycoplasma pneumonia may be present in the respiratory tract concomitantly with other pathogens. The radiographic findings in Mycoplasma pneumonia can be extremely variable and mimic the radiographic findings are extremely variable and mimic a wide variety of lung disease, but sometimes the radiological appearance coupled with clinical manifestation give clues to Mycoplasma etiology. In some patients, the organism may persist for weeks, 2 months in lower respiratory tract following acute pneumonia. Mycoplasma pneumonia can be very severe in otherwise healthy people. Although a few case reports of Mycoplasma pneumonia infection in pediatric AIDS patients have apparent in literature, and there has been speculation about possible role of Mycoplasma pneumonia. Mycoplasma pneumonia is inhibited by tetracycline, macrolides, ketyloids, and fluoroquinolone, with little variation among clinical isolates. Thus, in vitro susceptibility testing is not indicated for routine patients management purposes for infections of respiratory tract. Additional agents that are active at bacterial ribosomes, such as streptogramins, chloramphenicol, aminoglycosides, may also show
in vitro inhibitory activity against Mycoplasma pneumonia. Due to the lack of a cell wall, all mycoplasma are innately resistant to all beta-lactam and glycopeptides. Sulfonamides, trimethylpurine, and polymyxin, nalidixic acid, rifampicin are also inactive. Linezolid is the prototype agent of the oxazolidine class. These agents are much less active against Mycoplasma pneumonia. Fluoroquinolone have been shown to be bactericidal for Mycoplasma pneumonia. The extent of naturally occurring clinically significant acquired antimicrobial resistance for Mycoplasma pneumonia to any drug class is not known, but historically has been believed to be uncommon. التحضير مرة واحدة يا عبد الله، هذا هو مرة واحدة في اليوم. إذا ما حضرت اليوم حضر، إذا حضرت خلاص. Strains resistant to tetracycline and erythromycin have been isolated but are still believed to be uncommon in the United States. Recent studies evaluating macrolides, tetracycline, and fluoroquinolone indicates comparable in vitro activities against isolates obtained from North America and Europe. Since organisms from acute Mycoplasma pneumonia infection are seldom sought by culture, they are almost never tested for vitro susceptibilities for clinical purposes. Epidemics of Mycoplasma pneumonia infections occur in the community and in closed or semi-closed settings, military bases, hospitals, religious communities, unlike endemic disease, which has no seasonality outbreaks in the United States. Although the majority of infections are mild, Mycoplasma pneumonia is the leading cause of bacterial pneumonia. The incidence of Mycoplasma pneumonia is the greatest among school-aged children and decline after adolescence. Rates of disease among children under 5 years old have been reported to be low, but these studies were conducted at a time when out-of-home child care was less common. Mycoplasma infections are frequently spread among family members. In a recent study in Netherlands, 15% of family members of Mycoplasma pneumonia positive index patients had organism isolated from their nose. Transmission of Mycoplasma pneumonia usually requires close contact, such as in a family residence or an institutional settings with the high density of people. Transmission is believed to be by respiratory aerosol or by droplets. In view of intimate contact needed for droplet transmission and slow 6 hours generation time of Mycoplasma pneumonia, 1 up to 3 weeks incubation of each case is typical. In families, several cycles of infection may be necessary before intra-family transmission is complete. Some studies have been reported incubation periods from common source throughout as short as 4 days. Mycoplasma pneumonia is a mucosal parasite primarily involving the upper and lower respiratory tract, but it has been isolated or demonstrated by PCR in other sites, including urogenital tract, cerebrospinal fluid, synovial fluids, pericardial fluid, and cutaneous lesions. In post-mortem examination, Mycoplasma pneumonia has been recovered from the blood and the heart and the pericardial sac, spleen, brain, and kidney. However, frequency of direct invasion of these sites is unknown because the organism is rarely sought for clinical purposes. Mycoplasma pneumonia can be isolated from the pharynx 1 week prior to onset of clinical illnesses. The organism has been recovered as late as 4 months after the onset of illness, and for a much longer period in hypogamma globulemic patients. Attachment of Mycoplasma pneumoniae to the respiratory epithelium by its specialized attachment originally is a prerequisite for later events that culminate in production of disease. The close interaction between Mycoplasma and host cells protect the close interaction between the Mycoplasma and host cells protect it from removal by the host mucociliary clearance mechanism and allow it to produce variety of local cytotoxic effects. احيانا يا اخوان اوقف علشان اقرا بعض الرسائل او تجيني احيانا مسج من الادمن، احيانا اوقف مثلا دقيقه، نص دقيقه، دقيقتين للرد، فمعلش اذا توقف عندكم، لكن الشبكه قويه يعني ما في اشكال في النت عندي وان شاء الله ما يكون عندكم يعني. رابط التسجيل موجود يا اخوان، اللي بيسجل واللي ما سجل يسجل الان. التحضير. The tip structure is actually a network of adhesion interactive proteins and adherence accessory proteins that cooperate structurally and functionally to mobilize and concentrate adhesions at the tip of the organism. احنا بنحاول نضغط شويه يا اخوان علشان نخلص على 9 ان شاء الله، 9 وربع بالكثير. صلاة العشاء بنحاول نضغطها شويه. Once this polar structure is established in an independently assembled complex of protein B and C, the P1 is drawn to the structure to complete formation of functional terminal attachment originally. Attachment has been studied in detail in hamster model and in tracheal organs. An important area of research is whether the intracellular infection of host cells by Mycoplasma pneumonia and other mycoplasma is an important factor in the pathogenesis. لا، تسجيل الفيديو دكتور عبد السلام موجود عند الادمن، تطلبها منهم يا اخوان. The elements of host innate immune system are activated by Mycoplasma pneumonia, and attendant inflammation accounts for many of the initial signs and symptoms accompanying the early stage of evolving infection. Upon reaching the lower respiratory tract, the organism may be organized by complement or antibodies. Therefore, it is possible that complement plays a significant role in inhibiting growth of organism, particularly after inflammation-induced infiltration by inflammatory cells and oxidation. Adaptive immunity by both B and T lymphocyte has a major impact in the progression of Mycoplasma respiratory disease. ايه دكتور عبد السلام، مشيئة الله الباوربوينت ان شاء الله البي دي اف في اخر الكورس اطلبوه من الادمن يعطوكم اياه ان شاء الله، ما في اشكال، الله يحييك. But immunity is often short-lived and humans are susceptible to repeated infections despite the development of complement fixing and growth inhibiting antibodies response. Immune response that develop after infection often fail to eliminate the organism, implying that adaptive immunity can have a limited effect on the clearance of the established infection, thus allow asymptomatic carriages. Host response to Mycoplasma pneumonia range from asymptomatic to pharyngitis, bronchitis, bronchiolitis. The onset of Mycoplasma pneumonia is usually insidious, in contrast to abrupt onset of classic pneumococcal pneumonia. The symptoms include chills, headache, sore throat, malaise, cough. Cough is characteristically non-productive and irritant. at onset and may be paroxysmal. In young children, it may be staccato character and be exaggerated at night. Wheezing is common. دكتورة زينب والله إن شاء الله ما في إشكال، بس أنت تتابعي مع الأدمن، لأن أنت إذا فتحتي الزوم اسمك يكون موجود. Only few rails may be here despite an extensive infiltration. The chest film and the radiological infiltrate is more often unilateral than bilateral. Dullness is rarely present in examination of the chest since consideration is seldom complete. Epidemiological clues may be helpful in making the diagnosis of mycoplasma pneumonia because of pneumonia of other etiologies is infrequent among persons of 15 and 29 years of age. Likewise, a high proportion of pneumonia encountered in the summer is due to mycoplasma pneumonia because of other respiratory agents are then at low ebb. Isolation of mycoplasma pneumonia from respiratory secretions, throat, nasopharynx swabs of individuals with a classic respiratory signs is diagnostic, although serological or other confirmatory tests are useful. Treatment with tetracycline, macrolides, and ketyloids shorten the duration of illness. Because of the evidence of an immune component is, in some cases, mycoplasma pneumonia, CNS disease, and because of the benefits of steroid therapy and many CNS syndromes, steroids are frequently used in combination with antibiotics to treat mycoplasma pneumonia CNS infection. A 2002 review of steroids therapy in mycoplasma CNS cases suggests that it is often beneficial, particularly in severe cases. Another report documents successful recoveries of patients with permanent CNS disease that received a high-dose steroid therapy early in the course of disease, leading to suggestion that steroid therapy be initiated early in the course of the illness. Although full recoveries are reported, several cases document patients who relapsed with the clinical signs following cessation of first round of steroids. Interest in development of vaccine mycoplasma pneumonia has been evident since the 1960s. Soon after, the organism was successfully isolated in culture. This interest was fostered by lack of natural protective immunity following primary infection, prolonged carriage, and propensity for outbreaks of infection of military camps, schools, and hospitals. Animal models have been used extensively to improve understanding the natural immune response to mycoplasma pneumonia. Considering that attachment and initiation of local damage at the cellular and subcellular level are responsible for mycoplasma pneumonia disease, the logical vaccine strategy is to prevent attachment and thereby prevent initiation of disease. Development of serum antibody response after mucosal immunization may be or may not occur depending on the immunogen. Live attenuated vaccine have never made to a human use due to concern over residual virulence of the vaccine strains of mycoplasma pneumonia. Similar problems were encountered with temperature-sensitive mutant vaccines. Other vaccines candidates have included acellular proteins and polysaccharides component and recombinant DNA. The importance of P1 adhesion and mediating mycoplasma pneumonia, CETA adherence and initiation of disease is clear, but animal studies using P1 as vaccine antigen have not demonstrated protective efficacy. Experimental animal studies involving mice, hamster, guinea pig, chimpanzee have continued, but to our knowledge, there have been no recent clinical trials in human with any newer version of mycoplasma pneumonia vaccine, and no such trial appears to be further coming soon. Clinical trials of new antimicrobial agents for community-acquired pneumonia, such as the new of macrolides and equinolones have rarely included specific procedure for mycoplasma detection. Consequently, a better test of cure is needed to develop optimal antigenic antibiotic treatment therapies for mycoplasma pneumonia. The pathogenesis of extrapulmonary syndrome attributed to mycoplasma pneumonia infections need further investigation. Are such syndromes related to direct invasion or immunologic sequelae? It seems likely that either mechanisms may occur. PCR offers promise as a tool for antibiotic trials and studies of extrapulmonary syndrome, as well as a more rapid diagnosis, but methods need to be standardized and made commercially available for widespread use. Several products are already sold commercially in Europe, but none are available so far in the United States. Considering not only the respiratory disease caused by mycoplasma pneumonia, but also the many complications, vaccine seems desirable and should be encountered. It is important to realize that mycoplasma pneumonia infection can present as a very wide variety of clinical manifestation. The radiological findings of mycoplasma pneumonia can be extremely variable and mimic a wide variety of lung disease. But sometimes the radiological appearance coupled with the clinical manifestation can give clues to mycoplasma etiology. The inflammatory response elicited by mycoplasma pneumonia causes interstitial mononuclear inflammation in the lungs that may be manifested radiologically as bronchopneumonia. Multiple reports suggest that mycoplasma pneumonia infections are associated with exacerbations of asthma. Although this finding is not universal, the more important question is whether mycoplasma are a primary cause of asthma or if mycoplasma infection is a cofactor in the asthma development. Multiple lines of evidence suggest that mycoplasma pneumonia may play a role beyond simple and acute exacerbation. Mycoplasma pneumonia can be recovered more often from airway of patients with chronic stable asthma than from match-controlled patients. Mycoplasma pneumonia can be recovered more often from the airway of patients with a chronic stable asthma. Similarly, bronchial hyperactivity to methacholine challenges was present in 50% of patients 12 weeks after recovering from mycoplasma pneumonia compared with none in controls. Mycoplasma pneumonia is known to induce a number of inflammatory mediators implicated in the pathogenesis of asthma. زينب تحضير امس ما هو معي الان صراحه ما اعرف، بس اذا انت حاضره امس اسمك يكون موجود في الزوم انك دخلتي. Confirmed culture result can usually be available within 2 up to 5 days exclusive of specimen transport time and shipment, and if an off-site reference laboratory is used, uriplasma can be positively identified to genus level by their colonial morphology and Urase protection. Considerable attention has been given in recent years to the application of PCR assay in primary detection of mycoplasma and uriplasmal perinatal infection. Theoretical advantage of PCR assay for detection of genital mycoplasma include the fact that no viable organisms are necessary. Its analytical sensitivity is much greater than culture, and the result can be available in 1 day. There has been interest in the development of serological assay for the detection of the host immune response to genital mycoplasma. No assay for genital mycoplasma have been standardized and made commercially available in the United States. Any question, أخوان؟ أي سؤال؟ محمد، سامح، عبد السلام، المجيد, thank you all of you. سلمان, thank you very much. مشعل, thank you. ريان, وائل, نوال, فتون, نورة, بشرى, محمد, علي, أحمد, هديل, إيمان, ابتهاج, رحمة, وليد, محمد, لما, سمير, عبد الله, أسماء, فتون, تماضر, زياد, أحمد. طيب يا إخوان, thank you very much. We will move to the next presentation. وبعدها ناخذ بريك للصلاة بين تقريبا 8:10 إلى 9:15. وبعدين آخر محاضرة تكون بعد البريك. نخلص إن شاء الله 9:10 إن شاء الله. مصعب الصوت أتوقع عندك المشكلة يا مصعب ترى. طيب, our next presentation about pneumococcal infection. The pneumococcal pneumonia is a major cause of bacterial pneumonia and meningitis worldwide. The 8th edition and a large number of microorganisms may cause acute inflammation of the lungs, pneumonia, typical primary pneumonia. Pneumococcal pneumonia is generally preceded by a simple acute viral infection of the upper respiratory tract, such as a common cold, and it may also occur as a complication of influenza. It has been called captain of the men of death and also the friend of the aged, because it had long been the principal immediate cause of the most frequent contributing cause of the death and aged and infirm those with otherwise fatal disease. Prior to introduction of pneumococcal conjugate vaccine, pneumococcus was also the most frequent cause of acute bacterial meningitis in those over age 3 months. This remains true today in countries where conjugate pneumococcal vaccine has not been introduced, being exceeded in frequency only should a sporadic meningococcal epidemic occur. Prior to the introduction of pneumococcal conjugate vaccine, and this remains true today in the countries where conjugate pneumococcal vaccine has not been introduced, being exceeded in frequency only should a sporadic meningococcal epidemic occur. In the pre-antibiotic era, pleural epidemia was one of the most common complications of pneumococcal infection and still remains a common and increasing problem. In some studies, the pneumococcus is the most frequent cause of empyema occurring as a complication of either pneumonia or of the otitis media at all ages. And one pediatric cause of empyema occurring as a complication of either pneumonia or otitis media. In adults, an increase in the incidence of pneumococcal pneumonia empyema may be occurring in association with concomitant HIV infection and in children, exact reasons for the increase are not entirely apparent. In both adults and children, this increase appears to be occurring particularly in association with serotype 1S pneumococci. Less frequently, pneumococcus may cause focal infection at other sites of the body. It is also most common organism culture from blood of patients with acute febrile illness, usually pneumonia or meningitis, but not infrequently also from infants and young children with no demonstrable localized site of infection. Less frequently, the pneumococcus may cause focal infections at other sites of the body. It is also the most common organism cultured from the blood of patients with acute febrile illness, usually pneumonia or meningitis, but not infrequently also from infants and young children with no demonstrable localized site of infection. Morphologically, the pneumococcus typically appears as an ovoid or spherical coccidio-like form of 0.5 up to 1.25 nanogram in pairs, the distal ends of which tends to be pointed or lance-shaped and surrounded by a distinguishable capsule that contains polysaccharides. Diplococci may occur in chains, particularly in liquids medium. Hence, the designation Staphylococcus, the chemical composition of the polysaccharide determine its specific serological type, of which there are now at least 91. The diplococci usually stain gram-positive, but may decolorize and appear gram-negative in old cultures or after exposure to antibacterial agents. Resistant pneumococci to chemotherapeutic agents were recognized as emerging during therapy with ethyl hydrocodone, and which was first chemical to receive extensive clinical trials in pneumonia. Resistance to other antibacterial agents has been reported from many areas and had accounted for therapeutic failure. Reports have been included resistant to sulfonamide, tetracycline, erythromycin, and lincomycin. The most common route of transmission of pneumococci is by droplets from cases and carriers. In hospitals, the most frequent spread is from one patient to the patient and the adjacent bed. The disease rarely skipping a distance from more than 2 beds. Physicians, nurses, attendants, and others with frequent and close contact with patients are subject to colonization or infection from patients unless they take special precautions to avoid or reduce exposure to the patient droplets. Although pneumococci may be cultured from the air or from the dust or floor or the rooms for patients with pneumonia, they remain viable in the dried secretions for a long period of time.
Documented infection from such sources are rare. In cases of pneumococcal meningitis following skull fracture, the infecting organism comes from patients' own nasopharynx or accessory sinuses. And these foci are probably the source of primary pneumococcal meningitis. Subfractures may be associated with recurrent pneumococcal meningitis that no longer recurs after the defect in the cryptiform plate that has been repaired. As the root of infection is directly from the nasopharynx, it is possible that a serotype replacement in the conjugate vaccine era may be associated with more evidence of the replacement. Otitis media in the acute form and probably also acute sinusitis usually occur during or after simple upper respiratory tract infection. The incubation period of pneumococcal pneumonia is not known. Most sources state that it is 1 up to 3 days. It has been suggested that it is a few hours to about 48 hours. Indigenous infection in person who was previously asymptomatic carrier, then developed invasive disease can occur with incubation period of weeks. While significant details of human immunity to the pneumococcus are unknown, the exact mechanism by which it multiply in tissues and produce local inflammation or invasive disease remain to be fully elucidated. Pneumococci attach to respiratory epithelial cells by interaction with cell surface receptors. Mouse, rabbits, rats, and dogs are probably also human, can remove large number of inhaled virulent pneumococci. This is done in part by ciliated epithelium of the trachea and bronchi, but in greater measures by phagocytic cells. It is known that damage to the ciliary system by alcohol or other inhaled toxic substances such as cigarette smoke or by minor viral respiratory infections is necessary to establish pulmonary infection by pneumococci. In the dog infection occurs in the local alveoli and the pneumococci appears very rapidly in the local lymphatics, then in the hilar lymph nodes, then in the thoracic duct and subclavian vein, and finally in the peripheral blood. The healthy mouse, in which a single virulent pneumococcal injected intranasal instillation of the broth culture will also produce pneumonia and death in the mouse and in the rabbit. A dermal pneumococcal infection has been produced by intradermal inoculation in rabbits. Pneumococcal infection in humans are manifested in various ways. These range from healthy carrier state to minor respiratory infection or pneumonia and meningitis. Bacteremia may occur in association with manifestation ranging from simple pharyngitis and fever without demonstrably localized lesion. Pneumococcal meningitis is most commonly a complication of skull fracture, sinusitis, mastoiditis, or pneumonia. Clinically, pneumococcal lobar pneumonia begins characteristically most often after a few days of mild upper respiratory symptoms. If the condition is untreated, the organism may invade the bloodstream and consolidation spread to involve the entire root. It may spread to other parts of the lung with stormy course that ends in death or in recovery by rapid drop in the fever and cessation of symptoms. In many of these patients, pneumococcal pneumonia is the presenting feature of HIV infection, such that it has been recommended that patients presenting with invasive pneumococcal pneumonia should be offered counseling and testing for HIV infection. Investigators have studied the clinical presentation of bacterial pneumococcal pneumonia, comparing HIV serotype and HIV seronegative cases, and have noted some differences in presentation. While some differences were noted in the routine laboratory parameters and the microbiological data, however, recent analysis in which adjustments were made for age and severity of illness suggests that HIV-infected patients do, in fact, have a higher mortality, which is related, at least in part, to their level of immunocompromise, as indicated by their CD4 cell count. For many years, the treatment of choice for pneumococcal infection remained penicillin, to which the organism remained exquisitely sensitive. Prospective study of invasive pneumococcal infection involving 844 isolated from 22 countries on 6 continents documented no adverse effects of this therapy on the outcome of these infections. The study did document the negative outcome in cases with cephalosporin-resistant isolates treated with cefuroxime at dose of 750 milligram TID. More recently, several studies investigating the impact of antibiotic therapy on outcome of bacterial pneumococcal infection have suggested the outcome of such infection have improved in the use of combination antibiotic therapy. The reason for the benefit of combination therapy on outcome is uncertain and will be multifactorial in origin. Although controversy about the efficacy of active immunization with polyvalent vaccine persists, it has been mainstay for the prevention of invasive pneumococcal infection. However, investigators have also used passive immunization with specific intravenous immunoglobulins preparation in attempts to decrease the mortality of critically ill patients with severe pneumococcal infections without apparent benefit. Because of extremely high incidence of serious pneumococcal infections among South African workers, in 1911 immunized minors with a crude whole blood pneumococcal vaccine. Subsequently, a number of other investigators conducted clinical trials of the safety and efficacy of polysaccharides vaccine against pneumococci of various types. 2 different hexavalent vaccines subsequently were commercially produced and marketed. At about the same time, antimicrobials that were effective against pneumococcal became available in outcome of patients with pneumococcal infection improved substantially. Furthermore, most of the patients were either elderly or had chronic illnesses, which represented the population. Vaccine was tested with prospective double-blinded randomized controlled trials in young South African patients. This group of subjects was chosen because of extraordinary high incidence of pneumococcal disease they experienced, which enabled these trials to be conducted expeditiously and at a relative low cost. Despite the relative large number of subjects, the results were inconclusive because of low incidence of invasive pneumococcal infections. Small clinical trials conducted among patients with sickle cell disease found excellent efficacy of these patients, but the study was flawed because the trial was neither randomized nor blinded. In the United States, federal government surveyed over 12 months period in 1935 that involved 2.5 million people, and it was 558 cases of pneumonia per 100,000 patients. There were 3 illnesses diagnosed as pneumococcal over 3 years period. And in early 1980s, the CDC estimated rate of 100,000 patients per year of pneumococcal pneumonia is 68 up to 260 of pneumococcal meningitis and 1.2 up to 2.8 of pneumococcal bacteremia. Higher rates of pneumococcal infection were observed in patients with sickle cell anemia, congenital asplenia, renal transplant, Hodgkin disease, and multiple myeloma. On the basis of recent cooperative studies, the pneumococcal was found to be the most common pathogen cultured from respiratory tract infants. Penicillin has historically been antibiotic of choice for most cases of pneumococcal infection. The patient involved was 25 years old with hypoglobulinemia and bronchitis, who had received many courses of antibiotic treatment in the past. Until 1980s, there were only sporadic reports of strain intermittently resistant with MEC of 0.1 up to 1 nanogram per ml of penicillin. These strains generally had resistance to multiple antibiotics, including tetracycline, streptomycin, and others. Only small number of pneumococcal serotype have been associated with high level resistance. Generally, the level of resistant penicillin correlates with the resistance to all semi-synthetic penicillin and cephalosporin. Although low number of serotype dominate reports of invasive disease, and both children and adults during early part of 20th century, over the course of century, a higher number of pediatric serotype became more common, reflecting perhaps shift from direct contagion. For many years, the treatment of choice for pneumococcal infection remained penicillin, to which the organism remained excessively sensitive. A prospective study of invasive pneumococcal infection involving 844 isolates from 22 countries. The study did document negative outcome in cases with cephalosporin-resistant isolates treated with cefuroxime at a dose of 750 milligram TID. More recent several studies investigating the impact of antibiotic therapy on outcome of bacteriemic pneumococcal infection have suggested that outcome of such infection is improved by the use of combination of antibiotic therapy, most commonly macrolide and azalide. I think that was the last question. Any question, يا اخوان؟ أي سؤال؟ بالله يا اخوان اللي ما يحضر يحضر يا اخوان، لانه حنطلع البريك وبعدين حنرجع بعد الصلاة. نخلص قبل 9 ان شاء الله. شكرا هيام، شكرا فتون، سماح، عبد الله، ممدوح، محمد، مشعل، نجلة، معالي، صالحة، هيام، نوفا، ريان، ممدوح، سارة. Thank you very much all of you. محمد، فاطمة، سيد. باقي محاضرة واحدة يا اخوان. سلطان، باقي محاضرة واحدة بعد البريك ان شاء الله نخلص. محمد، عبد الجليل، جوهرة، دكتور عبد المحسن، جيا، تماضر، أحمد، محمد، أنفال، نورة، بندر، إفهام، نورة، أدهم، وضحة، سمير، سلطان، عبد الله، حنان. طيب يا اخواني، اذا ما في اسئله طبعا احنا نوقف الان لصلاه العشاء. معلش يمكن بعض الناس ما هو وقت الصلاه عندهم الان، لكن اي طبعا التحضير يا اخوان، لا تنسون التحضير، لا تنسون التحضير يا اخوان. المحاضرات بعد ما نخلص بكره ان شاء الله تاخذونها PDF من المنظمين يا اخوان. KH لا اسمك ما هو موجود يا دكتور. رمز. انتبهوا يا اخوان لا يكون اسمك رمز. شكرا على الدكتور عبد المحسن محمد. اللي يبغى يعرف اسمه يطلع على البارتيسيبنت المشاركين ويضغط عليها ويشوف اسمه يا اخوان، يتاكد انه اسمه مكتوب وليس نيك نيم او حروف. فرح موجود. ايوه ايوه صحيح عبد الله، من السن التاخير، لكن طبعا يعني نعطي اي اسمك كي اتش يا دكتور، فاذا بتغيره غيره يا دكتور. تهاني الرويلي والله ما اعرف يا دكتور، لانه في يمكن حوالي 700 فما اقدر اطلع الاسماء يعني. بس هنا مكتوب بندر الكاري. اللي يسأل موجود دكتور تهاني الرويلي، ما اعرف يعني. تحت البارتيسيبنت تحت يا دكتور تلقاهم 683 في الداشبورد عندك. طيب يا اخوان ويل بي باك. نراكم على خير بعد نص ساعه تقريبا او ثلث ساعه، ونكمل اخر محاضره ان شاء الله. اللي ما حضر يحضر يا اخوان، اللي ما حضر يحضر رجاء علشان نقفل بدري اليوم. Welcome back again. Assalamu alaikum. We will start our last presentation for today. It is about healthcare acquired bacterial infections. The term nosocomial is derived from the combination of Greek nosos, disease, and common as to take care of. Nosocomial is hospital. These infections are not present or incubated on admission, with exception that nosocomial infection may be present on admission if it is directly related to its residual or previous hospitalization. As a greater number of individuals are cared for non-acute care health facilities, understanding of the infection in these settings is also vital. Although we will continue to use the term nosocomial to refer to infection occurring, especially in the hospital settings, healthcare acquired is increasingly accepted as more inclusive term to describe infection that occur in the hospital and another healthcare settings. It is estimated that over 5% of all hospitalized patients fall victim to this infection each year in the United States. Since microbiological sampling, in clinical situation is neither routine nor uniform, the criteria are not dependent on laboratory for data for fulfillment. In addition to an HSN, some statewide incidence and prevalence studies have been reported in the United States. Over the past several decades, clinical microbiology laboratory has assumed an ever-increasing critical role for ongoing management and control of healthcare-associated infectious disease. The present and future needs for laboratory-based surveillance will require reliable detection of a new pathogen that emerge as a cause of important healthcare-associated infections. التحضير يا اخوان لو سمحتم بالله، هنخلص يمكن خلال تحضير اليوم الاول مو موجود عندي والله يا اخوان لكن عفوا هذا تحضير اليوم، تاكدوا بالله يا اخوان انكم حضرتم. This evolving rule dictates the need for strong collaboration between hospital and epidemiologists. The relative frequency of species of bacteria causing healthcare acquired infection has changed remarkably over the past 30 years. Bacteria that normally reside as a commensal on the internal and external surface of the host have been increasingly recognized as a cause of nosocomial infection. These bacteria then can serve as a source for infection in the colonized patient or are transmitted between patients nosocomially. The emergence of antimicrobial resistance has threatened to render the existing antibiotic are nasal useless. Increasing antimicrobial resistance is of great concern, not only because of agents available to treat these resistant infections are limited, but also because the treatment of H. disease rely heavily on empiric therapy. Patients may be infected with bacteria acquired from either indigenous or exogenous sources. Microorganisms from indigenous source may appear to be by reactivation of some previous infection. The pathogenesis of nosocomial infection is determined by host and environmental factors. Following infection with agents that persist at indigenous sites, significant immune depression may result in reactivation and uncontrolled dissemination. Pathogenicity by nosocomial agents has not been related to the presence of resistant bacteria. The selective pressure of antibiotic use assures the presence of resistant forms available for transmission of the healthcare setting. As with pathogenesis, clinical features and morbidity are determined by the virulence of agents and dose, and route of transmission and the character of the host defense mechanism. Infections of urinary tract accounts for 1/3 of all nosocomial infections. The great majority of healthcare-associated UTI result from instrumentation of the urinary tract, usually catheterization. In the catheterized patients, however, concentration of microbes considerably less than 10⁵ can be shown to increase if untreated. Pneumonia appears frequently in patients in whom aspiration has taken place or in whom an upper airways bypass by endotracheal intubation. Culture andograms or other staining of the sputum are important in establishing etiological diagnosis. Drainage of the purulent material or disruption of the wound is considered a prima facie evidence of wound infection regardless of the presence or absence of bacterial isolates. Intravascular catheters in place for more than 40 hours should be investigated even in the absence of inflammation. Nosocomial infection of skin, soft tissue, gastrointestinal tract, urogenital tract, and other sites may be found. Routine continuous collation evaluation and reporting of this data by trained specialists using surveillance tools are recommended. Infection control personnel must collaborate with engineers, nurse managers, administrators, architects, and physicians before and after surgery. As with the structure, the design of equipment is important to its correct use, maintenance, and disinfection. However, attempts to modify these expenses by reuse of disposable equipment and multiple patients have been responsible for several outbreaks of nosocomial infections and are to be decried. Although various professional organizations have all issued statements and published reports relative to issue of mandated public reporting, much uncertainty exists regarding optimal approach. I think that was our last slide. Any question يا اخوان؟ أي سؤال؟ We are done for today. خلصنا اليوم يا اخوان. Any question يا اخوان؟ شكرا نوال، شكرا سنجي، عابر، غدير. تحضير اسالوا غدير، اسالوا الادمن. انا ما عندي والله التندس حق امس ما هو عندي. هنوف، خلود، زينب، هيام. شكرا ساره.
عبد الله thank you very much عبد الله. وعد، هتون، ريان، أمل، يارا، الأملاك، أحمد، مرشد، ريان، حنين، وعد، زينب، مصعب، مشعل، سلطان، أحمد، عبد المجيد شكرا، رائد، صالحة، طارق، قدير، نورة، محمد، بدر، مريم، نورة. Thank you all of you يا أخوان. Yes sir, that's true. That's true, yes. Any question يا أخوان؟ اللي بيمشي يمشي يا أخوان. We are done. احنا خلصنا اليوم. اللي بيمشي يمشي. شكرا لكم جزيلا يا اخوان. يعني الاسماء ما شاء الله كثيره، فاشكركم جميعا كل واحد باسمه. يعني اتمنى لكم التوفيق ونتمنى انكم تكونوا استفدتوا ان شاء الله. اللي بيمشي يمشي يا اخوان. You can leave. بس تاكد انك حضرت. Make sure that you make your attendance before you leave. تهاني thank you very much. رابط امس مو عندي يا اخوان. اسالوا الادمن. والله ما هو معي. انا عندي تحضير اليوم بس. التحضير مره واحده يا اخوان. التحضير مره واحده. الادمن اللي سجلتوا معاهم يا اخوان، اللي عندكم الواتساب حقهم يا اخوان. ما هو عندي صراحه. Thank you very much إلي. بسمة thank you. عهود thank you. نزار thank you very much. إيناس thank you very much. شكرا نسرين thank you very much. See you tomorrow إن شاء الله سيد. Thank you all of you. داليا، محمد، تركي، حمزة thank you.